Okay, so good afternoon, everybody, and welcome to the SWOG Fall meeting. This is the Lymphoma Committee meeting, and we're really excited to have everybody here. On behalf of Jonathan Friedberg and myself, Sony Smith, I'd like to welcome our speakers as well as the audience. Jonathan, do you want to give a few words at the very beginning?
I think it's an extremely exciting time for the Lymphoma Committee. I know I speak for all of us in lamenting that we can't do this in person, but the pandemic has not slowed the progress of our committee. As you'll see over the course of the session, we've had great accrual over the last several months and some very exciting new concepts. But we're going to start with a session kind of focused on new agents and a way forward in lymphoma research. And I'm going to turn that part over to Dr. Smith.
Great. So there's certainly a lot going on, and we do miss seeing everybody in person. But what we are going to do over the next several hours is we've broken up the committee meeting into several different parts. The first is going to be a mini symposium discussing some of the advances in terms of therapeutics in lymphoma. And we have 3 of our own committee members who will be speaking. Each one of them will have about 10 to 15 minutes to do their presentation, and then we welcome any questions. There are several ways you can ask a question. You can either put it into the chat or use the raise hand function, and we will be able to call on you so you can actually speak in the middle. After we're done with our mini symposium on new agents, we're going to move on to talk about 3 recent activations. So these are trials that are available to you, and we hope you will support and be part of the process as we move forward in lymphoma. After that, we'll move on to an ASH abstract. So this was put together by one of our committee members, Dr. Stevens, and you will get a preview of what was submitted and accepted to ASH. And then we will go over some of our active studies and where things stand in terms of accrual, and also give you additional information if there are any questions. So without further ado, I think I'm going to move on to our mini symposium. And it is my pleasure to introduce our very first speaker, Dr. Nilanjan Ghosh. He is director of the lymphoma program at the Levine Cancer Institute in North Carolina. And his topic, let's see if I can forward this, or who has the controls. Oh, here we go. Here's the agenda. So we will start with Dr. Nilanjan Ghosh, who will talk about the integration of bispecifics with other agents. We'll then go on to integration of epigenetic modifiers in lymphoma treatment, and finish up the mini symposium with an integration of antibody drug conjugates in lymphoma treatment. So I already gave Nilanjan's introduction, so I'm going to let you go ahead.
Thank you, Sony, for the kind introduction. So today, it's my pleasure to talk about the integration of bispecific antibodies with other agents in non-Hodgkin lymphoma and B-cell non-Hodgkin lymphoma. Next slide. So just a quick introduction, which I don't think is needed for our group, but CD20, CD3 bispecific antibodies redirect endogenous non-specific T cells to engage and eliminate malignant B cells. And these bispecific antibodies are convenient to use, and as compared to CAR T cells, they are available off the shelf. We have several early phase studies which have shown promising activity in relapsed refractory B non-Hodgkin lymphoma, both in aggressive and in indolent lymphomas, which I'll show you in the next. We'll summarize in the next slide. And that has sort of been the premise to go into the combinations. So this is a very detailed slide, but just to show that these single agents, for example, mosunetuzumab, which is probably the most furthest along, we saw ASH plenary presentation in 2019 by Steve Schuster about this, and you can see that as a single agent in indolent lymphomas has a 43% CR rate, aggressive lymphomas about a 20% CR rate, and overall response rate is higher. So there is certainly activity. There were several patients who were post-CAR T in this as well. The PFS in indolent and aggressive, both seem to be, sustained to a good extent, 70 to 80%. At the follow-up, which is shown over here, and importantly, you'll see through these bispecific antibodies, although there's potential for CRS and ICANS, the CRS does differ a little bit between the groups, between the different antibodies, but mostly low-grade CRS, and ICANS is really rare. As you can see, glofitamab is a little bit different. It has two FAB portions to bind to CD20 and then one for CD3. So, and it also has very good activity. There's a recent publication which has this single agent activity as well. So that, and then odronextamab and epcoritamab. I don't mean to go through the details of and it is a little bit difficult to interpret these as well, because many of these phase 1 studies were, dose escalation and then dose expansion. So, for example, in the odronextamab, I put in over here the 80 milligram dose. If you see at the lower doses, you typically get lower responses. But then many of these studies have finally identified the right dose to start with. And for mosunetuzumab and glofitamab, there is also step-up dosing, so that lowers the risk of CRS. So, in general, you're seeing in somewhere between 20, and it's very varied, but 20 to 60% complete response rate in aggressive lymphomas. And that is, I think, encouraging, especially knowing the kind of patients who went on these studies. And as you can see, CRS rates are pretty low. So with that, I'll go to the next slide, and we'll start talking. Can we go to the next slide, please? So we'll talk about the combination of mosunetuzumab plus polatuzumab, which is a study which we participated in, and we'll go through the data. This was presented at ASCO by Elizabeth Buddy. I presented this at EHA. So we'll go through these slides. And the next slide, please. So there's some preclinical synergy between mosunetuzumab and polatuzumab, which is shown over here. You can see, the tumor decrease in tumor volume with this, and this synergy, which was seen in vitro, was in mouse model, was then sort of the basis for developing this phase 1 first dose escalation followed by dose expansion. So today what I'll present is the dose escalation cohort of 22 patients for mosunetuzumab and polatuzumab. Next slide. So this is sort of the design of the study. As I said, that there is, there was step-up dosing for mosunetuzumab. So what you can see over here, initially 1 milligram, 2 milligram, day 1, day 8, and day 15. And then, the cycle 1, day 15 dose was escalated from 9, 20, 40, and then 60. And then finally the 60-30 dose, which means for cycle 2, day 1 was also 60 milligrams, followed by, from cycle 3, day 1, it was 30 milligrams of mosunetuzumab. Polatuzumab was given for 6 cycles flat, and it was 1.8 dose milligram per kilogram on day 1 of each 21-day cycle for 6 cycles. This is a limited treatment study. So there are several bispecific antibodies which are sort of indefinite till progression. What I like about many of the glofitamab and the mosunetuzumab studies is that there is a defined course to it. And so for patients who achieved a complete response, mosunetuzumab given up to 8 cycles, and then it stops. For those who get partial response and stable disease, mosunetuzumab is given up to 17 cycles. Polatuzumab, as shown here, stops at 6. So that's sort of the design of the study. Next slide. All right, so baseline characteristics. The median age is 70. If you look at the 22 patients who were there, most of them are aggressive lymphomas. There were 3 patients with follicular lymphoma 1 to 3A. All of them have good performance status. If you look at median prior therapies, that's 3. for these patients. A third of the patients had received CAR T cell therapy previously. And then if the last two I think are important, 82% were refractory to the last prior therapy and 91% refractory to anti-CD20 therapy. So that kind of gives us how refractory this population was. Next slide. So now we look at adverse events, and when we look at every patient had some adverse event. Obviously, treatment-related adverse events were seen in 86% of patients. Now, looking at grade 3-4 adverse events, about 50% of patients had grade 3-4. There were 2 patients who had grade 5 events. They were not deemed to be treatment-related by the investigators. And there was 1 dose-limiting toxicity. The most frequent adverse events were neutropenia and nausea, followed by fatigue and diarrhea. And like 13% or 14% of the patients had AEs leading to treatment discontinuation. Some a little more than a third of patients had AEs leading to dose modifications. So next slide. So now looking at adverse events of special interest. So a cytokine release syndrome was seen in only 2 patients, so 9%, and they were all grade 1. Obviously, no tocilizumab, ICU, vasopressors, none of that, because these were just grade 1 events. And this is mainly fever and happened very early and then resolved. Infections were seen. So you can see over here about 30%, 32% patients had some infections. Peripheral neuropathy expected with polatuzumab. About 14% patients had grade 3 or higher peripheral neuropathy. And then importantly, neutropenia. So about 55% patients had neutropenia and 41% patients had grade 3 or higher neutropenia. There was GCSF, which was allowed liberally on the study. So, there was actually no incidence of febrile neutropenia in the dose escalation cohort. Next slide. So now, importantly, responses. I know the numbers are small, but within that, we can see that if you look at all 22 patients, 68% response rate with a 55% complete response rate. When we look at the majority of the histology aggressive lymphomas, 63% and then 47% complete response rate. In the post-CAR T, very small numbers, so things can change. Obviously, but they fortunately are changing for the better. 57% and 28% or 29% in the post-CAR T in terms of complete response, and then follicular lymphoma, all 3 patients had a complete response. So the reason I say that it's changing for better is updated a version of this, including several patients, like almost 3 times the number of this cohort will be presented at ASH by Elizabeth Buddy, and you'll see much more favorable complete response in the post-CAR T cohort, and overall is really holding as well. So that's, I think, very encouraging, considering that we are thinking of doing the study in the post-CAR T population using this combination. So next slide. This is just a waterfall plot showing nice tumor reductions in most patients. We can move on to the next slide. And this, I think this is a very important slide, because remember, this is time-limited therapy. So the plus signs are basically when treatment is stopped. And you can see the green circles are CRs. So you can see, like, in most of these patients, many of these patients at least, that there is sustained complete remission after stopping all treatment. And in the second patient, there you see a triangle, a blue triangle. The patient has progressed, but I can tell you that even after progression, so this protocol did allow retreatment. There's significant duration between stopping treatment and progression. So this patient has been retreated and is now back in complete remission. So that's basically a median was not reported for this part, but a median will, and the median fortunately has will not be reported, I think, because many of these patients are going to continue to be in CR, although our follow-up is relatively short. Next slide. So just to summarize, safety is really good. Expected side effects like peripheral neuropathy are seen. I think the big one is neutropenia. For sure. And then, CRS can be seen, mostly fever and very low incidence, low grade. Efficacy is really good in this heavily refractory population, having, CR rate of 47%, and many of these CRs, most of these CRs appear to be sustained. And, the numbers are small, but you'll see much better data at ASH. And so looking forward to that. Initially, when for the dose escalation cohort, admission was needed for the initial dose, but now that the CRS rate is so low that there's no mandatory hospitalization for the expansion cohort. It's also being tested in mantle cell lymphoma, relapsed refractory mantle cell. Next slide. So just wanted to bring up, this is just one glimpse of a combination, but let's look at all the combinations which are out there. And you can see, it's been combined with PD-L1 inhibitor, atezolizumab. There is also Mosun CHOP, and then Mosun CHP POLA, knowing that, it's heading in that direction. We'll wait to see at ASH the data for the POLARIX study. And then, it's also been combined with lenalidomide. There is also a TIGIT inhibitor, tiragolumab, plus minus atezolizumab, and I'll show you one slide about that. That's an upcoming study. And then, glofitamab is also being combined with all these different agents. And so combinations of bispecific antibodies with ADCs, with checkpoint inhibitors, and some with chemo is really being done. There's also a phase 2 or phase 3 randomized study being planned, a Mosun POLA versus R-GemOx. And there is also glofitamab with GemOx. And then if you look at the next slide, there's also combination studies with the other bispecific inhibitor, epcoritamab, and with different chemo combinations, and also with lenalidomide and relapsed refractory follicular lymphoma. So next slide. And I think that's going to be sort of the last slide. So there is a study of this Mosun plus tiragolumab plus minus atezolizumab. There's actually a previous very brief report of glofitamab plus atezo, which has been presented a couple of years ago, I think at ASH, but very limited data with very few patients. But this is a TIGIT antibody. And these T cells are found to express these molecules, like obviously we know about PD-1 a lot, but TIGIT is also in that same class. And by blocking this, you can enhance the anti-tumor activity of natural killer and T cells. So combining these with the bispecific antibodies is sort of trying to enhance the immune response against tumors. So next slide, I think that's pretty much it. Is sort of our future directions. So, as we think about these things, I thought, we'll just, what is the best partner for a bispecific antibody? Is it ADCs, chemo, IMiDs, cell mods, checkpoint inhibitors? How do we sequence bispecific antibodies, antibody combinations as a treatment option? In post-CAR T relapse, we are looking at obviously PR or SD after CAR T, not a candidate for CAR T. So there are patients who may not be able to get a CAR T cell therapy for whatever reason. Off the shelf treatment like this may be helpful, and then as a bridging therapy perhaps as well. And then the treatment duration, there are studies with limited duration treatments, and there are studies with more indefinite treatments where the chemo component or whatever finishes early, but then the bispecific continues for indefinitely till disease progression or adverse events. And then how do you monitor? Is there a better way than imaging to monitor these patients? We saw a recent publication in JCO with circulating tumor DNA in CAR T cell, both pre and post being predictive. So, trying to move MRD into an assessment, which may be, sort of easier than continuing to do scans and things like that. So I think that's it. We can go to the next slide. I think that was just questions. Yeah. So
that's great. Thank you so much. I think we have just a minute or two for questions. And, it's really exciting to see everything that's going on in this field. Maybe this is a question to the panel or to you specifically, but are you seeing a difference in response rates between rituximab refractory and relapsed patients, considering it targets CD20?
So, one thing is there that these studies mostly took patients who were CD20 positive. For example, recently we have struggled a lot to put patients in the dose expansion cohort because they're Rituxan refractory, but when we biopsy them, they don't have any CD20 detectable. So many of the Rituxan refractory patients have lost that expression or it's masked, and they haven't gone on the study. So that's something which we've recently seen, and so I can't tell exactly whether, how that is going to pan out. But you saw that in terms of most recent prior therapy, there were 80% patients who were refractory to most recent prior therapy. I would imagine a lot of that includes Rituxan plus something else. So, so that's certainly included. I don't know in that other 20% whether there was a big difference. We'll see if that comes out in the ASH presentation. So,
great. I think Jonathan has a question, and then we have one in the chat.
Yeah, real quick question. Very nice overview. It's striking to me that some of the data are, certainly with your combination, are rather similar to CAR T outcomes, and that the CR patients seem to be the ones who do really well. And it's maybe about half, a little better than half of people who are responding to the treatment. I know it's a small number of patients, but do you have any hints on predicting who's likely to respond and who isn't? Because you're committing them to a long, probably expensive treatment. And I'm just curious what efforts have gone on across maybe any of these to predict who's going to respond and who isn't.
Yeah, I wish we had. I don't think there's any predictors for response yet to this. I think it'll be this. If someone's immune system is really beat up, they may not. At least that's a hypothesis that they may not be the best candidates to respond to these bispecific antibodies targeting CD3. We'll get much more information at ASH. With much larger number of patients, seeing that that this is get more confidence in the post-CAR T cohort. I think the interesting thing which I brought up with Genentech is, are you able to detect CAR T cells there? Because the number of post-CAR T cells has expanded significantly in the expansion cohort. So is CAR T cell detectable at the time of response, or is it just endogenous T cells? Are we redirecting CAR T cells to fight against the tumor? That's an interesting thing, but the correlates have not been done. They did do interferon gamma and T cell activation markers. Although it was seen, and I didn't show that slide, although it was shown that both were present, those did not, or both went up, they did not correlate with responses. So I think that was something which was a bit unfortunate, because they were trying to correlate those, and it didn't really pan out. At least not in the 22 patient cohort. Whether it does in the bigger cohort, I don't know yet. I haven't seen the final oral, but we only have the abstract so far.
And then just very quickly, maybe one sentence from the audience. Do you feel comfortable treating these subjects totally in the outpatient setting?
Yes, absolutely. Yeah.
Okay, great. Thank you so much. We're going to move on to, and thanks to the audience for their questions. We're going to move on to the second talk in the mini symposium. This is Integration of Epigenetic Modifiers in Lymphoma Treatment by Dr. Jennifer Amengwal, who is an R01 funded physician scientist at Columbia University and a member of our committee. And with no further ado, go ahead.
All right, thanks. Next, please. Next slide. Thank you. Okay, very brief overview. Many of you know epigenetics, but this is a mechanism for regulating gene activity independent of DNA sequence. It's made up of really three general components. Writers, which are enzymes that add both histone or DNA modifications, such as P300 and CBP, EZH2 or DNMT. Erasers, enzymes that remove histone and DNA modifications, such as HDACs and demethylases. and readers, proteins that bind to histones and DNA modifications and alter gene activity. And some examples of these are bromodomains and SWOG and SWOG complex. And these work together to regulate gene activity without actually changing DNA itself. Next slide. Epigenetic drugs have been in development for more than 20 years, with our first approval of azacitidine and MDS in 2004. And as you can see, as the years have gone on, we've started targeting different targets between DNMT inhibitors, HDAC inhibitors, EZH2 inhibitors, and the list sort of goes on, more recently with BET or bromodomain inhibitors, PRMT5 inhibitors, and IDH1 and 2 inhibitors. However, next, sorry, I'm trying to click myself. Only 4 epigenetic strategies are approved, or drugs are approved in lymphoma. HDAC inhibitors are mostly approved for cutaneous T-cell lymphoma, with belinostat also being approved for PTCL. And there's potential applications in germinal center lymphomas, follicular lymphomas, and sarcomas, which some of this we'll talk about. And tazemetostat, an EZH2 inhibitor, is approved for follicular lymphoma and sarcomas, with also some potential application in other diffuse large B-cell lymphomas and T-cell lymphomas. So we'll focus on some of these applications in B and T-cell lymphoma in the setting of DNMTs, HDACs, and EZH2 inhibitors, and other novel ways to target these modifications. Next. Okay, so let's talk about diffuse large B-cell lymphoma. Recently, the cell of origin has been further split up into smaller and smaller slivers. And this is in part by the LymphGen classification, and there's been several papers that have similar clusters of gene expression profiles that have been identified in patients. Next, the GCB subtype could be split up in between 3 different clusters: SD2, MYC positive, and MYC negative. And all 3 of these gene clusters have frequent epigenetic mutations in genes such as TET2, EZH2, CREBBP, KMT2D, and P300, to name a few, as well as members of the SWOG complex, which leads us to believe that perhaps we can somehow target these mutations with epigenetic drugs. And this is now under intense study now that we have a little bit more closer look at which genes are mutated. Next. In addition, multiple chromatin modifying genes are altered in follicular lymphoma. As you know, follicular lymphoma and some germinal center lymphomas are basically originate from the same cell. And next slide. More than 96% of follicular lymphomas harbor at least one chromatin modifying gene alteration, and 76% of patients have more than 2 chromatin modifying gene alterations. And you can see these are very similar to those that are seen in diffuse large B-cell lymphoma. Next. Dysfunction epigenetic modifiers is also common in T-cell lymphomas, and this is a little bit more enriched in those from T follicular helper subtypes such as angioimmunoblastic T-cell lymphoma, and the mutations that we see most commonly are TET2, DNMT3A, and IDH2. So let's talk a little bit about this biology. When DNA is methylated, the abbreviation for that is 5-MC. TET2 can lead to hydroxy methylation of DNA, and this then sets the stage to allow for demethylation. Next. When there are mutations in TET2, there is decreased hydroxymethylation, which then leads to sort of an enforcement of over-methylation of DNA in the promoter regions. Next. Similarly, IDH1 and 2 lead to the conversion of isocitrate to alpha ketoglutarate and then to 2-HGG. When there are mutations in IDH1 and 2, next, we see that there's increased activity of IDH1-2, which then leads to an increase of 2-HG and blocks TET2, again leading to increased DNA methylation. And this is the biology behind why epigenetic therapies may have an enriched effect in angioimmunoblastic lymphomas with TET2 or IDH1/2 mutations. Next. So what's happening in the germinal center? Essentially, naive B cells enter the germinal center and they upregulate EZH2 and BCL6. And the reason for this is that these both lead to transcriptional repression. This allows for somatic hypermutation and class switch recombination to occur basically unchecked from other checkpoint sensors to allow for apoptosis. In the normal situation, as these cells travel through the germinal center toward the light zone, they're supposed to downregulate EZH2 and BCL6, increase IRF4 and PRDM1, and exit the lymph node as a memory cell or plasma cell. But in the oncogenic state, mutations build up in EZH2 and BCL6, as well as MLL and histone acetyltransferases and BCL2, and these coalesce to lead to a germinal center-derived neoplasm. Next. So can we integrate epigenetic therapies to resensitize lymphoma to treatments? We talked about a bunch of different mechanisms leading to epigenetic derangements in lymphomas, such as increased promoter hypermethylation, genome-wide hypomethylation in DNA, histone methylation and acetylation changes, and also changes in the SWE/SNF complex, which is basically slides the DNA along the histones. And so there's many different strategies to sort of reverse these abnormalities. And these abnormalities can lead to effects in expression of tumor suppressor and oncogenes, cell cycle, autophagy and protein degradation, immune surveillance, and apoptosis. So let's talk about some of these strategies, and we'll start with DNMTs. Next. So DNA methyltransferase inhibitors work by eliminating methyl groups in the promoter region. When there's overmethylation in the DNA promoter region, transcription factors can't bind, and this leads to inactive DNA. Next. So DNA methyltransferase inhibitors allow for the removal of methyl groups from the promoter regions, and then the ability of transcription factors to bind and activate expression. Next. Azacitidine and decitabine are approved for MDS, and they have common side effects such as cytopenias, GI disturbances, asthenia, and fatigue. Next. How does this fit with lymphoma? Well, it's found that DNA methylation, increase in heterogeneity of DNA methylation is linked to sort of more aggressive state in B-cell lymphomas, with the least amount of DNA methylation heterogeneity in follicular lymphoma and increase in activated B-cell, like diffuse large B-cell lymphoma. Next. And these methyl groups are not random. There seems to be an enriched modulation of DNA methylation in genes such as BCL6, MYC, and EZH2, which are key regulators of these types of B-cell lymphomas. Next. In addition, increased heterogeneity of methylation has been linked to an increased chance of relapse and a shorter progression-free survival. Next. And that increased methylation of SMAD1 was also linked to refractoriness in diffuse large B-cell lymphoma in those treated with R-CHOP. And DNA methyltransferase inhibitors have been found to sensitize diffuse large B-cell lymphoma to chemotherapy. So a combination of Dacogen and doxorubicin here really led to enhanced cytotoxicity in models of diffuse large B-cell lymphoma. Next. So based in part on these findings, a phase 1 clinical study looking at azacitidine plus R-CHOP chemotherapy was performed in patients with diffuse large B-cell lymphoma, where the overall response rate was 100% and a 92% complete response rate. It was found to be relatively safe with some reactivation of Hep C and neutropenia, but this was something that kind of sparked the interest of many investigators to pursue further investigation. Next. In addition, azacitidine is effective in T-cell lymphomas. Azacitidine studied alone in 19 relapsed refractory PTCL patients was found to have an overall response rate of 53%, with an enriched response rate in AITL, not surprisingly because many of these patients harbor TET2 mutations. And in part from these findings, a clinical study combining azacitidine plus CHOP in T-cell lymphoma as upfront therapy was pursued, where patients received oral azacitidine, which is CC486, as pre-treatment prior to CHOP chemotherapy. Next. And you can see that the overall response rate was 75% across the board, with a higher response rate in those with T follicular helper subtypes. Next. So based on these studies, there are now 2 active clinical trials through the cooperative group network that are combining azacitidine plus CHOP chemotherapy. A frontline study in PTCL, which we'll talk about later, looks at CHOP or CHOEP plus azacitidine in untreated T-cell lymphoma patients. And through the SWOG cooperative group led by Dr. Brem, azacitidine is being paired with mini-R-CHOP for diffuse large B-cell lymphoma in the elderly. Next. Okay, so other ways to alter methylation. EZH2 leads to increased histone methylation. Increased histone methylation leads to a condensed chromatin state and decreased transcription. And there are activating mutations in EZH2 in about 30% of germinal center-derived B-cell lymphomas. Next. EZH2 inhibitors work by blocking histone methyltransferase. Next. And this then enforces decreased methylation of histone, allowing for transcriptional activation. Tazemetostat is a first-in-class EZH2 inhibitor approved for follicular lymphoma. Next. It's also approved for sarcoma, and this is an oral medication with some mild side effects. Next. EZH2 has been linked to decrease MHC2 expression, and this is thought to impair immune surveillance in lymphomas. Next, and so in these patients that have high EZH2 expression, they have low MHC2 expression. Additionally, there's decreased infiltrating T cells in EZH2 mutated lymphoma samples in this mouse model, as compared to those with wild type EZH2. Next. And treatment with EZH2 inhibitors can reverse this, leading to re-expression of MHC2. Tazemetostat, as I mentioned, is the first approved EZH2 inhibitor. This is based on a phase 2 study, and you can see in this waterfall plot, most patients had experienced reduction in tumor burden. Next. It's well tolerated with grade 3-4 toxicities, including thrombocytopenia, anemia, and neutropenia. And you can see that there's activity both in mutated and wild type follicular lymphoma, as well as diffuse large B-cell lymphoma. And given the low toxicity profile, next, it's EZH2 inhibitors might combine nicely with other drugs to enhance the effects, especially in the setting where there are EZH2 mutations. And this is something we're trying to explore in the SWOG group. Next. Okay, EZH2 has also found a role in T-cell lymphoma, specifically ATLL or adult T-cell leukemia lymphoma, where there's overexpression, but no mutations found. And it's thought that the pattern is a little bit different and that there is a compensatory effect of EZH1, suggesting that dual targeting might be better. EZH2 overexpression is associated with inferior survival in patients with ATLL. Next. So based on these studies, there's a drug called valemetostat that's being studied in PTCL, and in the ATLL population, there is a greater than 50% response rate, substantial duration response, and a phase 2 is now enrolling. So we'll see how this pans out. Next. Okay, so finally, we'll talk about histone acetylation. I think this is probably most explored in B-cell lymphomas and T-cell lymphomas. Histone acetylation is catalyzed by the enzymes EP300 and CREB-BP, and there are loss of function mutations in about 40% of germinal center lymphomas. And this leads to when acetylation leads to an open chromatin state. And so when there's loss of function of histone-acetyltransferases, we again reinforce this transcriptional silent state. Next. You can kind of actually just go next until we get all the bullet points. Thanks. So histone-acetyltransferases, or HATs, lead to acetylation of histone, leading to an open chromatin state. We hear mostly about HDACs, which deacetylate histones, creating a chromatin state, condensed chromatin state. And although there are no mutations in HDACs, there have been found to be some overexpression in some lymphomas. And HATs and HDACs oppose one another. Next. Next. So HDAC inhibitors' mechanism of action is to, to block HDACs, letting HATs work unchecked, leading to increased acetylation of histone and transcriptional activation. As we mentioned, there are 3 HDAC inhibitors approved for lymphomas. Next, romidepsin, vorinostat, and belinostat all have similar mechanisms of action, target slightly different HDACs, but also target non-histone proteins, and they carry the side effect profile of GI disturbances and cytopenias most commonly. Next. So I mentioned HATs and HDACs oppose one another, and they influence both BCL6 and P53, which are very important in the germinal center lymphomas. CREBP, a HAT, is sort of in balance with HDAC3 and BCL6, and when CREBP is mutated, there's sort of a shift towards sort of an HDAC BCL6 phenotype, and using HDAC inhibitors may counteract this. Next. HDAC inhibitors lead to acetylation of BCL6, and this abrogates its effects, allowing for upregulation of LYMPH1 and allowing for cells to exit the germinal center with plasma cell differentiation. Whereas acetylation of P53 activates the tumor suppressor, then leading to activation of P21 and cell cycle arrest. Next. There have been many strategies to enhance HDAC inhibitors' effects in diffuse large B-cell lymphoma. We studied the combination of vorinostat plus a sirtuin inhibitor, niacinamide, and found that it was moderately effective, but we were able to demonstrate that we were able to acetylate BCL6 and P53 with this combination in mouse models. Next. In addition, romidepsin has been studied with chemotherapy in relapsed refractory patients, and this led to a modest overall response rate of 40%, and the PFS was lower expected with the toxicity greater than expected, so this has not really moved forward. Next. In addition, in the cooperative group network, Dr. Persky led a trial of SAHA plus R-CHOP chemotherapy in newly diagnosed stage 2 or bulky diffuse large B-cell lymphoma, and the progression-free survival was 81% with an overall survival of 90%. However, there were unexpected febrile neutropenia, and so this combination was not pursued further. Next. Similarly, combinations have been studied with HDACs and T-cell lymphoma. Romidepsin plus lenalidomide have been studied both in the relapse and frontline setting, and has had an impressive duration of response. In some studies, 8.9 months, which is higher than we see for most T-cell lymphomas. So perhaps there could be a role for this combination in the future. Next. We looked at the combination of romidepsin plus the novel antifolate pralatrexate and found an overall response rate of 71%. This combination was well tolerated, but had a short progression-free survival. And next. And also romidepsin has been studied in combination with the PI3 kinase inhibitor duvelisib, which led to an overall response rate of 50% and was fairly well tolerated. So some of these strategies continue to be studied. Next. We mentioned there's sort of modest effects of HDAC inhibitors across the board. Recently, there's a new HDAC inhibitor being studied, epigenetic, which does have an impressive response rate in the subtypes of lymphoma that we expect. follicular lymphoma, diffuse large B-cell lymphoma, and T-cell lymphoma. However, it does have a short median duration of response, only about 26 weeks. And it is associated with grade 3-4 thrombocytopenia. So new treatment schemas are being developed to see if this is something that could be tolerated and improve outcomes in lymphomas. Next. And some new data that's coming out is, can we perhaps directly target histone-acetyltransferases? Both CREB-EP and EP300 have sort of non-overlapping effects in different compartments of the germinal center. Next. So perhaps inhibiting HATs can lead to a synthetic toxic situation and cytotoxicity in lymphomas harboring these mutations. Next. In my laboratory, we're kind of taking the opposite approach, where we're looking to activate histone-acetyltransferases, taking advantage of the wild type allele. And this table represents the amount of drug needed to kill half of the cells. And you can see it's lower in those T-cell lymphoma cell lines and B-cell lymphoma cell lines harboring these mutations as compared to wild type. Next. We found that our drug can lead to prolonged survival in mice harboring these mutations. Next. And finally, we were able to reverse the decreased expression of MHC2 that's found in these lymphomas. Basically, our hope is that we can normalize the physiology here and eliminate B-cell lymphoma in that way. Next. So in conclusion, integration of epigenetic modifiers, is still a very active research endeavor here for lymphomas, especially in lymphomas originating from the germinal centers, such as follicular lymphoma, diffuse large B-cell lymphoma, and angioimmunoblastic, which may be most sensitive. HDAC inhibitors are approved for T-cell lymphoma, but have been combined with chemo and may be used in select subtypes of diffuse large B-cell lymphoma and follicular lymphoma. DNM team inhibitors have strong efficacy in angioimmunoblastic and T follicular helper T cell and may sensitize B-cell lymphomas to chemotherapy. And EZH2 inhibitors have enhanced efficacy in EZH2 mutant lymphomas, such as follicular lymphoma, as well as may have a strong effect in combination in diffuse large B-cell lymphoma and mature T-cell lymphomas. And with that, I'll take any questions.
Great. Thank you so much, Jennifer. That was really a tour de force of all the different things that we call epigenetic modifiers. I will open it up to the audience and see if people want to post things in the chat or if there's any other questions from our own panel. But in the meantime, I think we just have time for just maybe one question. One of the questions I always have about this field is, do you think it's more fruitful to combine different epigenetic modifiers with each other or with, other types of agents or like chemotherapy or immunomodulatory agents? Where do you see this field going?
It's a great question. I mean, all of those strategies have been explored. I think with single agent epigenetic targeting, as I showed you, many of these lymphomas harbor multiple mutations at the same time. And so they're able to sort of circumnavigate if we just target one epigenetic modification. So I do think combining with two epigenetic modifiers is a fruitful strategy, but also with other agents, these drugs can also upregulate CD20. They can do other sort of off-target activities that can enhance the effects of chemotherapy. And so I think all of those strategies are possible, but it's really utilizing them in the correct context in lymphomas that are sensitive to these drugs and not just across the board.
Great. Thank you so much. That was great. We're going to move on to the last talk of our mini symposium. So I'm going to introduce Dr. Shadman Mazyar, who is an associate professor at the Fred Hutchinson Cancer Research Center in the Clinical Research Division, where he focuses on aggressive lymphomas, CAR T, transplant, and today we've asked him to discuss the integration of antibody drug conjugates into lymphoma regimens.
Thank you. So my task today is to briefly review the class of drugs called antibody drug conjugates, or ADCs, and kind of to give a general overview of what we have today for lymphoid malignancies, and to kind of set it for discussion about how we can integrate these drugs for our current treatment landscape. Next slide, please. Next slide. So ADCs are not a new class for oncology in general. As you see on the right side, I saw this nice figure regarding all the ADCs that are approved for different oncology indications as of September, or I would say October of 21. For lymphoma, ADCs may be a relatively new class, but definitely an expanding one. And as I will show in the upcoming slides, we're having now access to a number of drugs that work with this mechanism of action. Basically, the idea is to take advantage of the cytotoxic quality of a chemotherapy drug and also the specificity of an antibody, and bring the chemotherapy agent to the cancer cell. That way, not only we increase the anti-tumor activity, we also reduce the toxicity because we're avoiding systemic exposure of that cytotoxic drug. Next slide, please. The MOA is pretty simple. Basically, we have a biological drug that is attached to an antibody, and the antibody circulates in the blood, attached to the chemotherapy or the anti-tumor agent, which could be a non-chemotherapy agent. And once it meets the target, which is a surface antigen on the cancer cell, it triggers internalization, and following that, we will have the release of the cytotoxic drug inside the cell, and depending on what's the mechanism of action for that chemo agent or anti-tumor agent, we'll have the cell toxicity. And as I mentioned, this would increase the efficacy, but also minimizes the systemic toxicity. Next slide, please. Now, there are a number of variables that are important in the design of an ADC, but also for us as clinicians to consider when use ADCs in clinical trials or in clinical practice. So we have the ADC structure on the left, basically simply an antibody. With the help of a linker, we attach it to the warhead, which is most of the time chemotherapy, and that creates the payload for us. So in terms of what target to use when we're designing the antibody, or when the antibody is designed, of course, the antigen has to be highly expressed on the tumor and not highly expressed on normal cells. We do need a surface display or surface expression of that antigen, and it does need to have the quality of being able to internalize the chemotherapy. Now, in clinical practice, of course, it does matter which antigen we're going for. And a good example and an important one in the current era is using, for example, CD19, which could be a common target for our approved CAR-T products, and also some of the recent, or one of the recently approved ADCs that target CD19. So, we do need to consider these points when we're using or designing studies. The antibody has to be specific to the target. The affinity is also important. The immunogenicity is important. The ADCs started with murine antibodies, which were just fully mouse antibodies, and then we moved to the next generations, starting with the chimeric antibodies, where we had the murine variable regions with the human constant region, and then we had the humanized antibodies, which were basically full antibodies, except for the CBRs, or the complementarity determining regions that were from the mouse. And then the third generation were basically fully human ADCs that we now have a number of, or a couple of ADCs that are approved from that generation. Linkers for lymphomas, most of the linkers are cleavable. In solid oncology, we have some ADCs that have non-cleavable linkers. And of course, the warhead is important because some of the toxicities that we see with ADCs is dependent on what chemotherapy agent is attached to the antibody. But of course, it has to be a small molecule and with a long half-life and stable at the time of circulation. Next slide, please. So the common warheads or chemo drugs that are used in combination with the antibodies for design of ADCs are listed here. I bolded the ones that are relevant to lymphoma, which is our topic today. And basically, from the 4 ADCs, 3 are FDA approved, 1 is not, but basically we either have brentuximab vedotin or polatuzumab vedotin, which use the monomethyl auristatin E or MMAE as the warhead, or with loncastuximab tesirine or camidanlumab tesirine. We use the PBD or pyrrolobenzodiazepine as the warhead. And these are basically the 2 kind of chemo molecules that are used for lymphoma ADCs, at least for now. Next slide, please. And I should mention that some of the toxicities, of course, are related to those molecules, and I will mention in the upcoming slides. So ALL and hairy cell leukemia are probably not, I mean, ALL is definitely not a lymphoma. Hairy cell leukemia has leukemia in the name at least, but being lymphoid malignancies, I thought I would give them 1 slide for both of them. So inotuzumab ozogamicin targets CD22. It uses caliciumycin as a payload, and it's approved for relapsed ALL. We won't talk more about it. And we do have a drug for hairy cell leukemia, which is moxetumomab pasudotox. This is another anti-CD22 immunotoxin antibody conjugate. So here, instead of chemotherapy agent, we have Pseudomonas endotoxin, and it is approved for hairy cell leukemia after 2 lines of therapy. Again, we won't spend more time. I just wanted to, for the sake of completeness, for lymphoid malignancies, mention them here in this presentation. Next slide, please. We do know brentuximab vedotin very well in the lymphoma world. Of course, this is antibody drug conjugate that targets CD30. It is a chimeric IgG1 monoclonal antibody, and it uses MMAE as the payload. It has a cleavable linker, and as we all know, it has FDA label for Hodgkin lymphoma from frontline to relapse post auto transplant maintenance, and of course for PTCL and in relapse and first line, and also MF as well. There was no really time or point of reviewing all the literature for brentuximab vedotin. It's relatively old news now, but basically based on Echelon-1 study, it is considered as one of the standard treatment options for frontline Hodgkin based on the modified PFS benefit in the randomized trial. In the relapse setting or after auto transplant setting, now multiple studies have shown great clinical efficacy, is now being combined with other agents for both relapse, for relapse and frontline setting outside of the BVAVD. And of course, and also post-auto transplant maintenance, the Atera study showed that in patients who are high risk by the definition of the study, there was a benefit in terms of progression-free survival. Same thing for PTCL with the first line and also in the relapse settings. But the point is, this is now a very well-established ADC that we're using in practice for years. And the first one for the lymphoma that kind of was added to our toolbox. Next slide, please. Staying on Hodgkin as a histology, we have another promising drug in trial. It's not, it's still considered to be investigational, and it's not FDA approved. Camidanlumab tesirine, and this is the last time I'm pronouncing it. It's Tami, we call it. It's a target CD25. This is a human IgG1. It's a 3rd generation ADC. Uses PBD as a payload, and again, a cleavable linker. It's not approved yet, but the promising study, as I kind of briefly summarize it here, for very high-risk patients with Hodgkin lymphoma with more than 3 lines, or if you count that auto transplant, or if not transplant eligible, more than 2. And these patients were, as you see, were almost 100% previously treated with brentuximab vedotin and immune checkpoint inhibitors. So very high-risk group. And we do see an overall response rate of around 66% with a CR rate of 27 or 28%, and with a relatively short follow-up, reasonable PFS and duration of response. Concerns, I would say, some PBD-specific side effects, which don't seem to be really limiting. Again, this is a very high-risk group, so risk-benefit ratio should be considered. And we know that there are some concerns regarding a number of cases of Guillain-Barré syndrome that were reported, but this is something that is being assessed. And again, risk and benefit in such a high-risk population will need to be considered. But a drug that, hopefully will benefit some of our patients with Hodgkin lymphoma is approved. Next slide, please. Some of the studies that are ongoing using Tami, and I listed them, and just the idea is that this drug is under development, and I'm sure there is to obtain the list of ISTs or investigator-initiated studies is not very easy. So I'm sure there are more studies that are ongoing with this drug, but at least to mention the few ones that are sponsored by the company, I kind of included them in this slide. Next slide, please. So moving on to diffuse large B-cell lymphoma, we recently had loncastuximab tesirine, or Lonca, as an FDA-approved drug for relapsed diffuse large B-cell lymphoma. This drug targets CD19, uses PBB as the payload, and has a cleavable linker, and it is currently approved for large cell lymphoma after 2 lines of systemic therapy. The study that kind of explains the rationale for this approval was the study that included patients with large cell lymphoma after 2 or more lines of therapy. You see the schedule, it's an IV drug every 3 weeks, although the dose for the first 2 cycles is different, but basically patients get every 3-week treatment for 1 year, and then every 12 weeks for up to 3 years. 145 patients were included with high-risk features that are listed there, and an overall response rate of around 50%, with a CR rate in quarter of patients, 25% approximately, and you see the median PFS and overall survival, as well as duration of response in responders and patients who achieved the CR. So again, drug approval based on the study, and as you expect with PBB, there are liver enzyme abnormalities. You do see some peripheral edema, and alkaline phosphatase increase. There were cases of pleural effusion, although the number was not higher than 20%, so it's not listed here. But again, an effective drug, and as I mentioned when I was talking about the targets for ADCs, this is an option. The question is how to time it and how to sequence it in relation to CD19 targeting CAR-Ts. It doesn't seem to be a major concern. At least there's no convincing data to show that. And this study, for example, they reported on 15 patients who moved on to CD19 targeted CAR, and they had a response rate of around 50%. I would say in general, both with this drug, Lonca, and also tafasitamab, which is another monoclonal antibody against CD19, which is not an ADC, and it's a naked antibody. We haven't seen so far much, any data that I know that showed that using those drugs before CAR-T would have a negative impact on the antigen expression, but this is something that we keep in mind. Next slide, please. Some of the ongoing studies with Lonca, as you see, both for large cell or high-grade lymphomas or low-grade lymphomas. One of the positive features of ADCs, given the limited toxicity being a targeted drug, is the ability to combine them to different type of backbones, chemotherapy, novel agents, and this is a nice example. For example, if you look at the LOTUS-7 study, there are a bunch of interesting combinations with other ADCs, polatuzumab in this case, with PI3 kinase inhibitors, with lenalidomide, and also chemotherapy, and venetoclax and other studies. Again, the list of ISTs, I'm sure it's not complete. For example, we are very close at our institution to have a protocol using this agent for auto transplant conditioning combined to standard treatment. So a lot of potential for these agents in different settings. Next slide, please. And polatuzumab vedotin is another ADC, which has been now approved for large cell lymphoma for, I believe, started with COVID 2 or 3 years, I believe. And so it's been used in practice. It's an MMAE, uses an MMAE payload, and It's approved in combination with bendamustine and rituximab for large cell lymphoma after 2 lines of therapy based on the study that was published at the JCO journal and was a randomized study with Pola v BR versus BR. And you see the PFS and overall survival curves there. Expected toxicities as we see with MMAE, and also the superiority of the responses and duration and long-term effectiveness. So we use this agent a lot in setting that is approved, but also as a bridging therapy before CAR T therapy is heavily used in our institution and other immunotherapy centers. Important drug. And next slide, please. And we just had the nice presentation by my colleague, Dr. Ghosh, about the polatuzumab study combined with mosunetuzumab. So, and great potential for combining this agent with pretty much anything as a backbone chemotherapy, novel agent, other ADCs. And this is just an example of some of the ongoing treatments. Next slide. And more importantly, we heard recently about, if you click one more time, please, the study that is a frontline study, POLARIX, which was a randomized study comparing R-CHOP to R-CHP with polatuzumab vedotin, basically replacing polatuzumab for vincristine. And we just had the press release that met the primary endpoint of PFS. We are eagerly waiting to see the data in form of presentation or publication. And if there is a very good chance, again, pending looking at the data and the toxicity, and it could be a practice-changing study and may bring ADC in the front line for a large cell lymphoma. Next slide, please, which is my last slide, I believe. So in summary, ADCs are an important class of drugs for lymphomas. We have a number of drugs for both Hodgkin and non-Hodgkin lymphomas. They have gotten to the first line for Hodgkin and most likely will get there for large cell lymphoma. Many studies are trying to get these drugs available for our patients with indolent lymphomas and effective drugs as monotherapy, and of course with combination therapy with the safety profile that's favorable compared to some of the other even oral targeted drugs, and basically very important drugs to consider when designing clinical trials in any settings, I would say. And with that, I'm happy to answer any questions.
That was great. Thank you so much, Maziar. That was really, again, just an incredible time to think about, a new class of agents, or actually maybe an old class with new payloads, I guess we could say, new targets and new payloads. I'm going to see if there's any questions from the audience or from our other panelists. And, if not, I know everybody is really excited about, the results of the POLARIX trial that will be coming up. One of the comments you made was about the ability to combine antibody drug conjugates with other chemotherapies. And yet, some of the payloads might have some overlapping toxicities. Do you have any thoughts about, particularly, I guess, the newest kid on the block, which has the PBD dimer, where that may be tolerated best, or which combinations look the most feasible?
I think maybe the nice thing is that if you look at the 2 ADCs that we have for large cell lymphoma, the approved ones, and the one for Hodgkin and maybe the upcoming one for Hodgkin, they use different payloads. So in a way, we have different antigens and we have different chemo drugs. So, there is an ongoing study, as I showed, using long-kind combination with Pola, which is basically delivering 2 different types of chemotherapy to do 2 different antigens. So at least right now, sounds like we don't have that limitation looking at the specific histology. As we get access to more ADCs, of course, as you mentioned, that would be extremely important to look at the safety profile and in combination with the backbone. I mean, some of the side effects could be overlapping there too. So a lot to consider before combining these agents.
Definitely the technology has advanced as well. I think that's very exciting to see. Great. Okay, we have one quick question. We do have 30 seconds here. So what was the, what were the number of cycles in the POLA or CHOP study? I think it was 6, right? It's the same as.
Yeah, I think it was 6, and I think both arms had 2 additional cycles of rituximab, I believe.
Okay.
So yeah.
Okay. Thanks to all 3 of our speakers, and thanks to everybody for joining the mini symposium we had. I think it really reflects all the great work that's happening in lymphoma, and certainly we're, as part of our group, integrating some of these agents into our new trials. So I'm going to turn now to recent activations. We have 3 studies we're going to have our committee members present. The first one we can pull up is, again, by Dr. Jennifer Amengwal, which is Alliance A051902. Go ahead, Jennifer.
Okay, thank you for inviting me to present. So this was just recently activated. This is a randomized phase 2 study of duvelisib, or 5-azacitidine, in addition to CHOP or CHOEP, in comparison to CHOP and CHOEP for CD30 negative T-cell lymphoma. Next. So just as way of background, PTCL is a rare heterogeneous disease, makes up only about 7% of all non-Hodgkin lymphoma, and there are 19 clinical entities here that have very varied pathologic presentations and outcomes. Median age of diagnosis is 65, and treatments have historically been derived from those used in aggressive B-cell lymphomas. Although different histologies have unique biology, which may inform future treatment options. The PFS is pretty bad. 5-year survival is about 20 to 30% for most subtypes. So, there's a dire need for improvement in upfront therapy for T-cell lymphoma. Next. So we spoke about this a little bit earlier, but recently there was a study combining oral azacitidine plus CHOP chemotherapy for patients with PTCL, NOS, including multiple different subtypes. 17 out of 20% of patients had AITL or PTCL NOS with a T follicular helper type subtype, and more than half were CD30 negative. This combination was very well tolerated with some cytopenias, febrile neutropenia, there was some thrush and strongyloides, but overall it worked quite well with an overall response rate of 85%, CR rate of 55%. And at the end of treatment, the overall response rate was 75%. This was enriched for patients with T follicular helper subtype. So patients were treated with oral azacitidine prior to starting CHOP, and then they basically had this sort of, not pre-phase, but sort of preliminary treatment, and then it continued on days 8 through 21 for each cycle. And as you can see, excellent responses. Next. Duvelisib is a PI3 kinase inhibitor, which has been studied in T-cell lymphomas and found to have really excellent activity. It's been studied using 75 milligrams BID for 2 cycles, and then 25 milligrams BID until progression or tolerance, and intolerance. And the overall response rate was 52% with a complete response rate of 36%. And there's other studies combining duvelisib with other active agents in T-cell lymphoma, which have shown similar toxicities and improved efficacy. There are side effects of duvelisib, mainly liver enzyme increase and cytopenia, some rash, edema, pruritus, and dizziness. Next slide. So based on these data, a trial is advised for upfront T-cell lymphoma in patients who are untreated and who have less than 10% of CD30 expression. And this is because brentuximab plus CHP has been very active and is now approved in CD30 positive T-cell lymphoma. So we need a strategy for those who are CD30 negative. The study will stratify patients for whether or not they're T follicular helper subtype positive, and patients can receive either CHOP or CHOEP based on age, and we'll stratify for that, as well as their age. So those over the age of 60 will receive CHOP, and those under the age of 60 will receive CHOEP. And basically, this will be a 1-to-1-to-1 randomization of duvelisib plus CHOP or CHOEP, azacitidine plus CHOP or CHOEP, or CHOP or CHOEP alone. And patients will be treated for 21 days. The first 12 patients in each arm will be part of a study check, and some correlative studies will be monitoring MRD status, gene expression profiling, PROs, and PET-CT evaluation. Next. So the primary objective is to look at complete response rate by PET scan. Secondary objectives are to look at toxicity and tolerability, as well as overall response rate, duration of response, PFS, EFS, and overall survival. We'll determine whether the designation of follicular helper T cell phenotype is correlated with response, and we'll assess FDG PET features and correlate that with response. Exploratory objectives will be to look at cell-free DNA, to monitor MRD, and to use gene expression profiling, immunophenotyping, and mutational analysis to try to distinguish between Th1 and Th2 profiles and their prognoses. Next. Eligibility is pretty simple. I'm not going to go through this in too much detail, but just want to stress again, this is for CD30 negative or less than 10% expression, new diagnosis of PTCL. Next. I'd like to talk a little bit about safety monitoring. So there will be a lead-in specifically for those with duvelisib CHOP studies, in that there hasn't been this combination study before. So in cycle 1, patients will be seen weekly, and then they will be monitored bi-weekly with safety calls to evaluate for emerging toxicities. They'll be followed with CBCs weekly. And the first 6 patients to be randomized to duvelisib and CHOP and duvelisib CHOEP will be evaluated very closely for DLTs. And if there are 2 more DLTs in cycle 1, accrual will be suspended and we will reevaluate this regimen. Next. In the phase 2 portion, there will be safety checks. If there's greater than a third of patients that develop toxicities within the first 2 cycles, the study will be paused. And some of these toxicities that we're looking for specifically is grade 4 febrile neutropenia, grade 3 thrombocytopenia with bleeding, grade 3 or greater non-hematologic toxicities, and a delay to starting cycle 2 or 3 for more than 7 days. And if this toxicity accumulates, then we'll have to decide whether to move on with the study. Continue. Next. So essentially, this is the schema for the 3 arms. As I mentioned, duvelisib will be given concurrently, orally, on days 1 through 21 of each cycle. Azacitidine will be given sort of starting at day -6 from cycle 1, and then continuing from day 8 to 21 for each cycle through cycle 5. And arm C will just be CHOP or CHOEP for standard care. DNA, blood samples will be collected for DNA on days 1, 8, 15, and cycle day 1. Next. Correlative studies, as I mentioned, we'll be looking at TFH phenotype by IHC, gene expression profiling for GATA2 and TBX21. We'll be looking at cell-free DNA by CAP-seq, and looking at PET scans, as well as quality of life through patient-reported outcomes. Next. There is a tissue requirement for this study. We'll be requesting 15 slides or 1 block to do these correlative studies. They could be 5 unsaved slides or other stained slides. And this is detailed in the protocol. The cell-free DNA will be collected in the Streck tubes. You can continue. And there'll be patient-reported outcomes to understand toxicity and potential curative therapy to make sure that this isn't overwhelmingly toxic. And this will be completed, hopefully, electronically. Next. Finally, there will be imaging that will be evaluated to really assess PET scan in the setting of T-cell lymphoma, which is not so well defined. This will be after cycle 4 and then after cycle 6 and end of treatment. Next. If you have any questions, please feel free to contact me or the PI, Dr. Nehamet Shah. And this is just open. I don't think there's any patients accrued yet, but we're hoping to accrue patients soon. With that, I'll take any questions.
Great. Thank you so much. This is really exciting to get, one of the first, frontline T-cell lymphoma studies going through the intergroup, and really hope that everybody who's on this call will, consider activating this. There is time for maybe just one or two questions. One is from Mark Hoffman. Do you allow auto transplant in CR1? Which I think you may have addressed. And then the second one is about the dose of duvelisib. How was that chosen, given that overall response and gamma inhibition is thought to be better, albeit with more toxicity at the 75 milligram dose?
Yeah, so transplant is allowed in CR1, and we'll see it. Most patients with PTCL at this time are advised to receive consolidated transplant following chemotherapy. The dose of duvelisib was, some debate. We'll see what happens, and we hope that it'll be safe. But this was, after lots of discussions. There is no phase 1 data combining duvelisib with CHOP chemotherapy, so there is a safety run-in to see if this will be tolerated.
Great. All right, thank you.
Sonia,
yeah.
If I could just make a general comment for people, I would really encourage people to open this trial. I think it goes without saying that as exciting as the B-cell world has been with new agents and improved outcomes, we really have not seen that in T-cell lymphoma. And previous randomized studies have been negative. It's really only the cooperative groups that can do such a trial with different types of agents and comparing them. And this is really only going to work if we kind of embrace this uniformly. I understand these patients are relatively uncommon, but they're out there. SWOG in the past did have success with an upfront T-cell lymphoma trial as far as accruals. So we're really looking to everybody. And I think although there was a lot of detail in Dr. Menguel's talk, in the end, this is a pretty straightforward randomized trial that once the safety lead-ins are complete, won't have a lot of extra visits or things like that for patients.
Very well said. I agree. Great.
Thank you.
Okay, we will move on to the next activation that is recent. This is ANHL 1931, which is nivolumab in combination with chemoimmunotherapy for the treatment of newly diagnosed primary mediastinal B-cell lymphoma. And I think we can move on to the next slide. It'll be Dr. Tysel Phillips, who is an associate professor at the University of Michigan, who will be presenting. So I think we need to move on to the next set of slides. Oh.
Yeah, I think these are the extra slides.
Here we go. Okay, this is the right one. He's presenting on behalf of Dr. Manali Kamdar, but it is Dr. Phillips and not Dr. Stevens. So go ahead, Tysel.
All right, thank you, Sony. So good afternoon. So on behalf of Dr. Kamdar, presenting the ANHL 1931, a randomized phase 3 intergroup trial of nivolumab in combination with chemoimmunotherapy for patients with newly diagnosed primary mediastinal B-cell lymphoma. Next slide. So primary mediastinal B-cell lymphoma is a rare subtype of aggressive non-Hodgkin's lymphoma, primarily affecting the AYA population. Current outcomes with chemoimmunotherapy, R-CHOP plus radiation has been historically recommended. Obviously, based on the patient's age and sometimes gender, radiation is something we sometimes try to avoid through long-term complications from radiation therapy. So dose-adjusted EPOC was something that was evaluated by the NIH. Their single center phase 2 study revealed a 5-year event-free survival of 93%. This was replicated in the pediatric community. They have a multi-center phase 2 study with a 2-year event-free survival of 69%. And from that paper, determined that it wasn't much better than historical treatments. And a multi-center retrospective trial had a 3-year event-free survival of 87%. All three, albeit with smaller numbers than what we typically used to seeing in some of the more common non-Hodgkin's lymphoma subtypes. Genetic alterations in the PD-L1 locus, especially 9p24.1, which leads to PD-L1 and L2 amplification, has been noted in primary mediastinal. Then PD-1 inhibitors in relapsed refractory primary mediastinal has shown some promising efficacy and safety. Next slide. So the rationale for the study is that we have a rare subtype of non-Hodgkin's lymphoma, primarily affecting the AYA population, without a truly defined standard of care per se. Intergroup collaboration is key to move the field forward. As mentioned before, current outcomes of chemoimmunotherapy, we have R-CHOP plus radiation, dose-adjusted EPOCH-R without radiation. The rates of dose-adjusted EPOCH-R may not be as high as what was demonstrated in NIH, and important for us as a medical community to validate the previously seen results in a larger prospective study with and without radiation. Genetic alterations in PD-L1 locus of 9p24.1 leads to upregulation and amplification of PD-L1 L2. Then we have responses with PD-1 inhibitors in relapsed refractory primary mediastinal. As such, it's important to test PD-1 inhibitors plus chemotherapy as frontline therapy. Next slide. So the general hypothesis is nivolumab combined with chemoimmunotherapy for the treatment of newly diagnosed primary mediastinal will result in a superior long-term progression-free survival when compared to just chemoimmunotherapy alone. Next slide. So this is a very busy slide, which is simplified in the next one, but in essence, we'll have 3 arms divided into 2 smaller arms. So arm A is dose-adjusted R-EPOCH by itself, then dose-adjusted R-EPOCH plus nivolumab. Arm C is R-CHOP by itself, then R-CHOP plus nivolumab, and then arm E is R-CHOP plus radiation, and then R-CHOP plus radiation plus nivolumab. So it's physician's discretion. The physician will declare the chemoimmunotherapy backbone for one of the following, the 3 that we mentioned. And as we see, the decision to use radiation should be mentioned up front. If not, it will be counted as an event unless a biopsy and disease is sort of determined. Next slide. And as I stated, the next slide is a bit more simplified. So basically, we have 3 cycles, a 1-to-1 randomization with 3 cycles of chemoimmunotherapy versus 3 cycles of chemoimmunotherapy plus a PD-1 inhibitor. Interim PET assessment, obviously progressive disease off therapy, an additional 3 cycles of therapy in those who are responding or have stable disease, and then an NF treatment PET scan. So those who are in CR who are randomized to chemo alone will get observation. Chemo plus radiation will get radiation at that point. Partial responses will be observational biopsy. Biopsy positive will get radiation. If you predispose radiation to begin with, you'll get radiation at that point. Stable disease, again, off therapy of chemo alone. And then if radiation was chosen, you'll get radiation at that point. Next slide. Again, it's the stated radiation is to be chosen above in the beginning. So the primary endpoint is progression-free survival is determined by independent review committee. Secondary endpoints, event-free survival, overall survival, rate of PET positivity, Duval score 4 to 5 at the end of treatment. Stratification will be 1-to-1. The backbone, either R-CHOP plus radiation, R-CHOP without radiation, dose-adjusted R-EPOCH. The age, since this is a pediatric population, they will also stratify by age, less than or over 18. There is an ability to receive 1 cycle of chemotherapy prior to enrollment, and which will be discussed in the next slide. You don't need to repeat imaging in those patients. And exploratory objectives explore the potential biomarkers predictive of response to treatment, including the 9p24 molecular alteration, baseline expression of PD-1, PD-L1, and circulating T cell subsets measured by flow before and after treatment. Next slide. So enrollment is greater than equal to 2 years of age at time of diagnosis. Patients must have histologically confirmed primary mediastinal as defined by the WHO criteria. Again, patients who enroll after completion of 1 cycle of clinical studies, including imaging obtained before the start of chemoimmunotherapy, will be acceptable. Next slide. So for the statistical considerations and accrual goals, you have an assumption of an 80% PFS in the standard arm with a 1-sided alpha of 0.05, the 80.7% power to detect a hazard ratio of 0.33 with Nivo with a PFS of 92.8% in the experimental arm. If the accrual is robust, they'll plan to convert to a 1-sided alpha of 0.025 With the original assessment, it'll be 186 patients, evaluable patients, which comes down to 62 patients in each arm, 31 divided if plus or minus radiation. Sorry, plus or minus nivolumab, 236 if the alpha is changed to 0.025. They anticipate an accrual rate of 4.2 patients per month, estimated accrual rate of 50 patients per year, including patients from COG, Alliance, ECOG, and SWOG. Again, estimated time to complete accrual will be 3.8 years. Next slide. So in conclusion, primary mediastinal B-cell lymphoma is a rare non-Hodgkin lymphoma subtype with increased incidence in a younger patient population. Treatment goals are improving outcomes while minimizing long-term toxicity. Cross-network collaboration will enable prospective trials moving forward. Next slide. And just acknowledgements for those who are involved in the creation of this protocol. And that concludes the slide deck.
Great. Thank you so much, Tyson. This is, a really incredible collaboration between all the groups that you have listed on that slide. There's one question from Hildy. Are any PRO assessments in this study?
So from the slide deck that I have from Anali, I mean, it didn't mention it, so there may be, but I'm just not sure from what I have.
Mm-hmm. Yeah. Jonathan, any comments at all? Or?
I think similar to the other one, this is an important study in an uncommon disease, but a disease that affects young people. And, we've made some advances, but this is a real precision approach, taking advantage of a genetic lesion. So it's a very rational study to do. And I think, again, one of the nice things about this is it's very straightforward. I think we're all comfortable using checkpoint inhibitors and, just kind of combining it with our standard approach. So although that first schema that Dr. Phillips showed looked a little bit overwhelming, this is really a pretty straightforward trial.
Yeah. And I was just going to add that, the ability to give one cycle of therapy before people start, I think is really important. I mean, we've learned along the way that not allowing that for the sickest patients or with the most rapidly progressive disease, can sometimes bias our results. So I think that's a really important part on a practical level for everyone who's listening. So, okay, great. Thank you so much. We're going to move on to the third recent activation. And this is by Dr. Elizabeth Brem, who is an assistant professor of medicine at UC Irvine, and wrote this trial with lots of hard work. But go ahead, Dr. Brem.
Yes, so I think the last time we presented this study, I got to tell you that it was freshly activated, and this time I get to tell you that we actually have patients enrolled, which is very exciting. So many of you have seen the development of this trial over the last several years. This is our phase 2/3 randomized study of our mini-CHOP with or without oral azacitidine in our patients aged 75 and older. Next slide, please. So I don't want to spend too much time on background because we've talked about this study over several meetings, and thank you to Dr. Amaguel for that wonderful setup in terms of the preclinical and early clinical rationale for this trial. But part of the reason we wanted to look at this population is that DLBCL is incredibly common in this older population, but they're underrepresented in studies, and there's really no clear standard of care. And for a number of reasons, the outcomes in this population are inferior to our younger patients. Again, Dr. Amaguel set us up beautifully with the rationale for why we're considering a hypomethylating agent in combination with R-CHOP. And the other thing we're really put a lot of effort to and we're very proud of in this study is the way we have incorporated geriatric assessments to help us understand our population and inform in the future, for example, who might be most likely to benefit from combination chemoimmunotherapy. Next slide, please. So here's the study schema. So we are, in terms of histology, so this is DLBCL. We are also taking grade 3 follicular lymphoma. You can have a transformed lymphoma. The only thing that is specifically excluded is a Richter's transformation from CLL. Transformed lymphoma from any other histology is allowed. And we are allowing double hit and triple hit since there is no standard of care, particularly in this age population. In terms of stratification factors, we're looking at the 75 to 79 crowd versus 80 plus. The rationale for that has to do with a lot of our mini-CHOP studies have been in those 80 plus, but in practice, it's often being used in younger patients. You can see the other stratifications there. So the key thing that's different, and I'll talk a little bit more about this, Sonia had mentioned that in many studies, we're trying to push to kind of have the ability to have some treatment, so we're not selecting for a sicker population. In this case, there isn't a cycle of R-CHOP or our mini-CHOP that's allowed prior, but everyone gets what's called pre-phase therapy, which in this case is going to be, in the first iteration of the study is prednisone and vincristine, and going forward is going to be just prednisone prior to randomization. And you can see here the randomization. Arm 1 is the CC486 or oral azacitidine with the mini-CHOP versus mini-CHOP alone. Next slide. And so similar to the T-cell lymphoma study that was talked about, we are doing a safety run-in. We do have some data looking at CC486 with full dose CHOP, but A, that was full dose CHOP, and B, there weren't a ton of older patients that were represented on that study. And so we are starting with a 40 patient safety lead-in with a 1-to-1 randomization between the CC486 or oral azacitidine versus mini-CHOP alone. And we had this lead-in be randomized because we know there can be toxicity and unfortunately even fatal events with our mini-CHOP alone in this patient population. We, as of this morning, have 9 patients accrued. So we are about a quarter of a way through our safety lead-in. Once we complete our first 40 patients, we'll have a pause until everyone has completed cycle 1. If we have no concerning safety signals, we'll move on to the randomized phase 2 component where the primary endpoint is 1-year PFS. And then if all goes well, then we'll move on to our randomized phase 3 with our primary endpoint of 2-year PFS. And you can see the accrual targets here. Next slide, please. So key eligibility criteria are for you. We've kind of addressed most of this. We tried to make this as user-friendly as possible. Any criteria are very specific. So for example, because we are using anthracycline, we felt like we had to have a reasonable EF cutoff, but we're hoping that otherwise this is fairly wide open and going to be relatively easy to accrue to. Next slide, please. So again, the original version of the protocol, just to go back to the pre-phase therapies, the original version of the protocol had people getting 1 dose of vincristine and 7 days of prednisone. This has proven to be a little bit difficult for a number of reasons, including kind of some variation in terms of pre-phases that have been studied where some use vincristine and some don't, the logistics of giving the vincristine, what day should the vincristine be given on. So ultimately the revision that was submitted to CTEP last week will be only prednisone. It will be 600 to 100 milligrams and 4 to 7 days, depending on the discretion of the treating investigator, basically how much debulking you think the patient needs. And you can start this prednisone at any time prior to registration. So you can, if you've got a patient who needs something, you can start that prednisone and then screen them and register them for the study. And the only other thing is we clarified that this pre-phase or the prednisone has to be given within 14 days of starting treatment on your assigned arm, whether that's your arm mini-CHOP or whether that's the CC486 or oral azacitidine arm. We realized that we actually never addressed CNS prophylaxis in the first iteration of this protocol. Patients now may receive up to 4 cycles of intrathecal therapy, but there will not be allowance for any high dose IV methotrexate. Next slide. Again, the geriatric assessments are something we think is really unique to this study, and there's 2 of them, just as a reminder if you're new to this study. So we have the S1918 Comprehensive Geriatric Assessment, which come from our colleagues in the Alliance Cancer Aging and Research Group. This is about a 20-minute assessment. It takes, there's provider components as well as patient components, and this will be done as 4 time points, as you can see here. And really the question we're asking here is, particularly by adding an additional therapy, what does that look like on patient functioning, not only in the short term, but also in the long term, hence the 12-month and 24-month assessments. Then there's another tool, which is called the FIL or Italian Lymphoma Foundation tool. This was recently published in JCO, and this is administered once at registration to assess frailty. So patients are assessed as either fit, unfit, or frail. The reason this is here is there was some evidence from this group, from the Italian group, that patients who are frail maybe are less likely to benefit from combination chemoimmunotherapy versus those who are fit or unfit. And this is administered once at the beginning, and this takes about 7 to 10 minutes. And I believe I have a QR code in the slide deck in terms of it should be very easy to access. You could do it on paper, you can do it online, whatever's easier for your site. Next slide, please. So again, just to kind of give you the take-home points about why you should be excited about this study and consider opening it if you're not already in the process, as I got a report yesterday, I believe, that we've got just under 50 sites up and running, but still many, many in the pipeline. So thank you to everyone for your enthusiasm so far. We really appreciate it. It's the first time we've ever looked at this population in the NCTN. We think the pre-phase for everyone is a unique feature and eliminates some problems for the study. We talked about how the first 40 patients are part of the safety run-in. Just logistically, should you open this during the safety run-in, we are having bi-weekly safety calls, and the patients do have to be seen, whether it be remotely through telemedicine or in your clinic once a week during the safety run-in. But after that, the number of assessments does drop significantly. So we hope that this is a fairly user-friendly study. Again, we think the geriatric assessments have the potential to select therapy for patients in the future. I didn't talk about them. Many of our other colleagues or other studies have talked about circulating tumor DNA assays. We are using them in this study, not only to look at absolute amounts of circulating tumor DNA, but also given the use of the azacitidine. And we talked about this a little bit at one of our prior meetings. Look at the methylation patterns in the circulating tumor DNA to help kind of understand how this drug is affecting things at a molecular level. And in general, we think that we have the potential in this study to change the standard of care for patients in multiple ways, given not only the therapeutic question, but the geriatric assessments and the circulating tumor DNA assays. Next slide, please. Or is this my last slide? There we go. So these are your QR code and your link, and please just email me. I'll send you anything you want to the FIL tool, which again, it's very user friendly. You can do it online, you can do it on paper, but basically it's a quick assessment of ADLs, IADLs, and some comorbidities. Takes about 7 to 10 minutes, and then it spits out a score of whether your patient is fit, unfit, or frail. And I think we might have just a moment for questions.
Great. Thank you so much. That was really great. And I just want to echo, I think the theme for the 3 recently approved trials is that each one of them are focusing on, really unique populations with some precision medicine approaches. And I think certainly for the older patient, what this trial is going to be really important to help us, try to get to a better cure rate. There's 2 questions in the chat for you. So one is from Boyu. Is there a reason for the eligibility criteria to have a performance status requirement of 0 to 2, given that we are trying to study frailty? And it also skews the IPI to be lower, and the stratification factors include IPI in it, which may also skew results.
This was a conversation that we had multiple times during the development of this trial, because the other part of the question here is not only are you skewing the population, but there's a lot of patients who might have a performance status of 3 the day you meet them, but it's from disease, and 3 months ago they were in a different spot. So ultimately, we did decide to put a cutoff of 2, really more from a safety issue, just thinking about that we do feel like we have to do this safety run-in, particularly for the oral azacitidine, and we didn't want to have a population that was going to be too vulnerable and open them up to too much toxicity. But I'll also say that, that's one potential advantage of this prednisone pre-phase, and also the fact that the prednisone can be given kind of prior to registration. So maybe I shouldn't be saying this, but you could maybe have a patient who has an ECOG of 3, give them a week of prednisone, and then they might be an ECOG of 1 a ZUBROD performance status of 1 to 2 and be able to enroll. So there is, and particularly if you think that this decrease in performance status is from disease, that might be something you could actually rectify potentially with the prednisone pre-phase.
Yeah, I completely agree. And that was a big part of the pre-phase being included. Last question, are you looking at death from lymphoma progression or chemo side effects versus death from existing comorbidities?
I don't know if we have explicitly discussed this. Certainly, I think this is something we're going to have to take into account, particularly if we end up having any deaths during the safety run-in, which, knock on wood, so far we haven't. But we have not explicitly discussed how we're going to look at the deaths. And so maybe that's something I need to go back to the stats folks and think about.
Yeah, I think we will be looking at cause of death, and we just need to include it or incorporate it in a better way. Okay, well, thank you to everybody for that portion. For the next part of our open meeting, I'm gonna switch over to Dr. Friedberg, who's going to introduce our next speaker.
Thank you. And we're excited to have Dr. Stevens present an ASH abstract, give people a little preview of what's going to be shown at ASH based on results from a study that we've published previously. So Dr. Stevens is at Huntsman in Utah and was involved in some of the long-term follow-up work that we had done on the early stage studies. And this is an important subset analysis.
Thanks, Dr. Friedberg. On behalf of my co-authors, I'm excited to present this abstract entitled Extranodal Presentation in Limited Stage Diffuse Large B-Cell as a Prognostic Marker in 3 Consecutive SWOG Studies. You can go to the next slide. As you all know in this group, limited stage diffuse large B-cell is defined as an Ann Arbor stage 1 or 2, which means that it is essentially localized to one lymph node group or organ or 2 lymph node groups within the same side of the diaphragm. Next slide. Recent trial results have aimed to minimize the number of chemotherapy cycles and/or eliminate radiotherapy without compromising the excellent outcomes seen in these patients. These studies have changed the standard of care for limited stage diffuse large B-cell, but you can see the NCCN guidelines here, which give several options for this patient population. However, there may be subsets where an abbreviated approach is really insufficient. Next slide. This research was partially stimulated by the publication of the Memorial Sloan Kettering's group, where they performed a retrospective study where they reviewed about 340 patients with stage 1 diffuse large B-cell lymphoma who were treated with R-CHOP for 4 to 6 cycles plus or minus radiotherapy. In their research, they had some key findings. They found that extranodal versus nodal presentation had shorter progression-free survival and overall survival. And they also found in those extranodal patients who received consolidative radiotherapy versus no radiation had prolonged overall survival. And this result was more pronounced in patients with a positive PET scan at the end of the chemotherapy portion of their treatment. So the aim with our research was to validate or dispute findings in this Memorial Sloan Kettering publication by analyzing patients with limited stage diffuse large B-cell who were treated on 3 consecutive SWOG studies, the S0014, S0313, and the S1001. Next slide. On these studies, enrollment occurred from the year 2000 till 2016, and there was a total of 276 combined registrations for the study. We did have to exclude some patients from our analysis, and you can see the reasons on the right-hand side here, including incorrect histology in 28 patients, most of which were follicular lymphoma, incorrect stage in 7, bulky disease in 2, and insufficient baseline documentation in 5, which left us with a total of 234 patients. And you can see across the bottom the number included from each study, with 60 from S0014, 43 from S0313, and 131 from S1001. And go to the next slide. There was some variation in treatments, but I've highlighted that in our evaluation, about a quarter of the patients got therapy with R-CHOP ×3 plus involved field radiotherapy plus Y90 or Zevalin. And then another quarter of patients received R-CHOP ×3 plus involved field radiotherapy, and the remaining half of the patients received R-CHOP ×4 cycles. You can go to the next slide. In terms of the baseline characteristics of the 234 patients in our analysis, the median age was 62 with a range of 18 to 85. A stage-modified IPI score of 0 to 1 was present in 68% of the patients, and 44% of our patients, or 104, had extranodal disease. We compared the baseline characteristics between patients with extranodal and nodal disease presentation, and we found that There were no baseline characteristics that were statistically significantly different between the groups, and that included age, stage, LDH, and IPI score, among others. I've included a table here to show exactly what extranodal sites were included in our presentation. And you can see the percentages on the right-hand side, and you see that the largest percentage of extranodal presentations came in the head and neck region at 44% of our population. And you can see the list of those locations that I included in that head and neck presentation. The next most common groups were our bone presentation in 13, skin, muscle, and soft tissue in 12%, and GI tract in 11. And you can advance to the next slide, please. For our patient population, there was a median follow-up of 7 years with a range of 1 to 16 years. I've highlighted here the progression-free survival and overall survival. And I think importantly, we have 10-year estimates of progression-free survival at 71% and overall survival at 77%, which are both quite good. And I'll also be highlighting some of the differences and similarities between our study with the Memorial Sloan Kettering group. And I think it's notable that we have exactly the same 10-year overall survival as their group at 77%. When you look at the progression-free and overall survival curves here on the right, you'll notice we don't have a lot of events out at the 15-year mark. But we have yet to define a very clear plateau in the progression-free survival curve, given that there are a few events out far. But this is similar to what we've seen in many other studies. It's also notable in our study that there were 50 deaths, and the most common known cause of death was lymphoma in 32%, and second cancer in 12%, with unknown and other comprising the remainder of deaths. Next slide. Interestingly, in our study, we actually disagree with what the Memorial Sloan Kettering publication showed, in that in our particular patient population, there was no significant difference in progression-free survival between patients with extranodal versus nodal disease. And you can see the overlapping curves here below. Go to the next slide. I listed here the survival based upon extranodal site. Please note that a lot of these groups are very small, so we were not able to perform clear statistical differences between these groups. But I did want to highlight the head and neck group because this is the biggest group of patients that we have. And actually, it does have a shorter 10-year progression-free and overall survival than many of the other groups displayed here. You can go to the next slide. Interestingly, also in contrast to what was found in the Memorial Sloan Kettering analysis, we had opposite findings that we did not find a significant difference in progression-free and overall survival between patients with extranodal disease treated with radiotherapy versus no radiotherapy. Go on to the next slide. We also specifically looked at the group of head and neck patients, since that's the largest part of our population, and found that there was no significant difference in progression-free survival between patients with head and neck extranodal disease treated with or without radiotherapy. We didn't have a lot of patients that had one PET analysis or two had a positive PET at the end of therapy. Only one of our studies mandated interim PET, and this was the S1001. And of the 55 patients with extranodal disease, only 5 of those patients had a positive interim PET scan. And we defined this as a Deauville score of 4 or 5. And of the 5 patients with a positive PET, all received involved field radiotherapy and 1 progressed. Go to the next slide. I really wanted to include a slide just to highlight differences because we're finding some different findings than what was published by Bobbio et al. This year. And so I wanted to highlight a few differences in our patient population that may or may not explain the differences in our findings. Our analysis did include stage 2 disease, and that was 41% of patients, whereas only stage 1 was included in the MSK study. There were more patients with bulky disease in the MSK study, and that was defined as a lymph node size of 7.5 or greater, and they had 10% in their study. There is a different distribution of extranodal sites between our two groups of patients. In the MSK study, the GI tract was the most common site of disease in 27%, followed by bone at 21%, and head and neck at 15%, which you recall in our group was 44%. Also, primary testicular lymphoma was included in their analysis and comprised 9%, and all of the studies from SWOG excluded testicular involvement. You can see the different treatment regimens listed here between the groups, and you can see that about a third of patients in the MSK study received R-CHOP times 3 to 6 cycles, and the remaining 2/3 had R-CHOP for 3 to 6 cycles plus radiotherapy. Interestingly, there was a little bit different median radiotherapy dose between the two groups, which is also listed here. You can go on to the next slide. And in conclusion, in our analysis, we found that limited stage diffuse large B-cell lymphoma had prolonged progression-free and overall survival regardless of extranodal versus nodal presentation and whether they received consolidative involved field radiotherapy or not, which is in contrast to the conclusions of the MSK study. Our data does not support extranodal disease as an adverse prognostic factor in limited stage diffuse large B-cell lymphoma, and as such, we do not routinely recommend radiotherapy consolidation in non-bulky limited stage diffuse large B-cell just because they present with extranodal disease. Of course, we didn't have enough patients on our study that had an interim PET to make a recommendation for PET adapted involved field radiotherapy in those with positive PET. The majority of our group had extranodal disease of the head and neck, which appears to have similar survival as nodal presentation. And as seen previously, there was a continuous rate of relapse without plateau of the PFS curves, and the most common known cause of death was lymphoma, which supports the need for long-term follow-up, and it also tells us that there may be patients that are not adequately treated with our frontline therapy. You can go to the next slide. And I just included the acknowledgements here for all the folks that contributed to this study. And I'm happy to take any questions.
That was really great. Thank you so much. I do want to particularly call out the contributions of Dr. Persky on this. He couldn't be on the call today because of a family issue, but he's been working in this space through that whole generation of trials that you presented, and was instrumental in helping to pull this together as well. A few good questions have come up, which I think it'll be a little bit of practice for Ash. I guess the criteria for describing bulk, it sounds like at Memorial that was 7.5 centimeters. Was our criteria 10, or how did you calculate that for those couple of patients you excluded?
Yeah, so Dan and I actually went through all of the patients and looked at the list, and we matched that to their criteria for this. So we just have there were very few patients with bulky disease, as most of the studies actually excluded that.
And the second question was about baseline genetic characteristics, which I don't believe we have a lot on, that trying to correlate that with extranodal versus nodal presentations.
Yeah, do you mean cell of origin or any specific genetic characteristics?
So let's start with cell of origin. I think we do have that on some of the patients.
Yeah, we do have cell of origin on some of the patients on some of the studies, but we didn't have enough to really provide any analysis here. We're going to go back through that probably before the publication and see if we can get enough patients to say anything meaningful about. And also most of the patients did not have FISH, except, we went through and did that on the S1001 study, and there were very, very few patients that had a MYC translocation. So I don't think we'll be able to really say much about that.
Years ago, many years ago, Tom Miller tried to do an analysis of gene expression in early stage versus advanced stage, and actually presented at ASH what he thought was a signature. In the end, it was probably more a signature of extranodal versus nodal. And of course, when you're doing gene expression, you're capturing stroma as well. So, if one wanted to do a deep dive now with sequencing, it could be interesting to try to understand whether there are true biologic differences or whether there's a lot of noise there, but the number of patients is very small. The other question that Dan had addressed previously based on the long follow-up, and we should emphasize that in these patients, many of whom are older, was about the timing of deaths from lymphoma versus deaths from either second malignancies or other causes. When the median age of patients coming into the study is in the mid to late 60s, and you're out at 10 or 15 years, there are going to be deaths that occur just naturally. And that may explain, and I think it's an important point as we make this point, that it may explain the lack of a plateau. And any thoughts on that?
We did, Hong Li just gave me some information about, I was just looking at the different extranodal presentations by, organ site and trying to figure out if those, deaths were more related to lymphoma versus other. And I couldn't find a clear pattern there, but we haven't looked at the whole group. And so I think that this will be a really great analysis to do for our manuscript.
Steve, did you have a question? No. Okay. I just saw you. Sony, anything you want to add?
No, I think, these questions that have been coming through are really going to be very helpful, not just to prepare for ASH, but also for the manuscript itself.
Yeah, I'll, just my, of course, fully biased here, but I think this is a very important contribution because the memorial paper, in a very memorial-like way, basically said, well, based on these data, we should be giving radiation to all of these extranodal presentations, which, as was demonstrated, is a reasonable percentage of these patients. And we're just not seeing a signal at all to make that recommendation. And I think getting this out there so people can make a decision on an individual patient basis will be important. All right, so do we know when this is being presented at ASH if people want to come and visit?
I believe it is Saturday night.
Okay.
Yeah, Saturday from 5:30 to 7:30.
Does anybody know are posters going to be in person as well as, I think they are, right?
Yeah. I think they're going to include both a video and a poster.
Everybody's submitting a video, and then you have the option to be in person.
Sorry, I missed that.
Everyone's submitting a video, and then if you are there in person, you'll be asked to be by your poster as well.
All right.
With your mask.
We hope. Come and visit if you're there in person. Show some support. Excellent job. Very nicely done.
That's great.
Thank you.
All right. So now we're going to do a rather quick kind of round robin summary in the last hour or so here of the ongoing trials. And we've really asked people to just present highlights and updates because most of these trials have been presented before. So I'll try to introduce each speaker. And the first one is the 1826 study, Dr. Alex Herrera.
Thanks, Dr. Friedberg. So as you all know, S1826 is our phase 3 randomized study evaluating patients with newly diagnosed advanced stage classical Hodgkin lymphoma. We're comparing brentuximab vedotin plus AVD compared to nivolumab plus AVD. Next slide. So this has been a collaboration across the cooperative groups, and I really have to say, as the study's gone on, it's been really, we're really grateful to all of you. SWOG has really led the charge, and I'll show the accrual data in a second. But also we've had great partners in COG and the other adult cooperative groups in North America. And we're waiting for our first Canadian patient, but the sites are activated, and hopefully we'll get one soon. Next slide. So this is the schema for the study. Patients are randomized 1-to-1 if they are determined to be eligible to receive either nivolumab plus AVD or brentuximab vedotin plus AVD for 6 cycles. They get their PET at the end of treatment. And then there are some criteria if an investigator opts to give radiation therapy at the end of therapy, though they have to declare that the intent to do that prior at the time of registration. Interim PETs are optional. They are required in the pediatric patients. So the sample size for the study in total is just under 1,000 patients, 987. The primary endpoint is progression-free survival. Our target enrollment had been 20 to 21 patients a month to fully accrue the study in 4 years, but I'll show you that we're ahead of that, which is really to all of your credit. As I mentioned, it'll be 6 cycles. I think the main thing to mention about the treatment is that G-CSF is required for patients who are randomized to receive brentuximab vedotin plus AVD, and it is optional for patients who are randomized to the nivolumab plus AVD arm. And in collaboration with COG, we've built in some, kind of a really nice patient-reported outcomes, kind of survey correlatives. Next slide. So as of a little over a week ago, we had almost enrolled 600 patients. And on the left, you can see that SWOG has enrolled almost 40% of the patients. So as I mentioned, really leading the charge. And I thank all of you profusely for your support of the study. COG has really done a fantastic job as well, though. They enrolled just over a third of patients. And the other cooperative groups have also been doing their best to enroll as well. And the kind of leading enrolling sites are on the right here. And as you can see, a lot of, primarily it's SWOG sites. So again, thank you all. As you can see, we're nearing 2/3 accrual of the study. You can go ahead to the next slide, which shows the accrual by month. And you can see that yellow dotted line is our target accrual, which is that, like I mentioned, like I said, 20 to 21 patients. And we've been well above it, even during the pandemic. So it's been really, impressive, and a credit to all of you. The trial is enrolling really well. There's been great enthusiasm for the study. We even had a month where we enrolled 54 patients. I thought we'd be done by now if we had stayed at that rate, but we came back down to the stratosphere, not quite down to earth. So next slide. We do have some updates. It is an international study, in Australia, New Zealand, and Canada are participating. There is an upcoming amendment that's going to add the FIL assessment, which you heard from Dr. Brem in her DLBCL study. Elderly patients with Hodgkin lymphoma do represent kind of an understudied group of patients, and they're allowed to enroll on this study, which is really a great opportunity to learn how they do with these regimens. So we're going to incorporate that, not the comprehensive geriatric assessment, but just the very short survey tool at the baseline at enrollment. And if patients who have been already enrolled, we'll ask that the provider do that retrospectively on the patients that have been enrolled. And then there'll be some small adjustments to the PRO battery. As we've gone on, we realized that there were some additional symptoms that we could ask about. That's all I have. Thank you again to all of you for your participation and support of the study.
So just one editorial comment, because this is regarding Dr. Brem's talk too, that we keep using this word elderly, and it's starting to be a real slippery slope when you're describing patients over the age of 60 as elderly. So maybe we'll just say older, which I think is the term we're supposed to use, but
Agree.
I will say that just hot off the presses that Rochester will be putting their 28th patient on tomorrow, because I have somebody who I've signed and we're going to register tomorrow. But it's going very well. I think. Any questions for Dr. Herrera? And if you're a site that hasn't opened this yet, there's still time to open it and meaningfully contribute. We estimate at least another 12 to 18 months of accrual. And I also want to thank everybody for their contributions on submitting the required blood and scans. We've had really impressive compliance on that, and it's going to be very meaningful as we try to dissect which of these drugs may help particular patients. All right, let's move on to the S1608 trial. Dr. Barr from Rochester.
Thanks, Dr. Friedberg. Alex is always a tough act to follow. Thankfully, he's actually part of this study too. I'll give you a quick update on the randomized phase 2 trial in early progressing or refractory follicular lymphoma. Next slide. And I've gone on about the background many times over on this one. Just to remind everyone, we're enrolling patients who are refractory to or relapsed within 2 years of bendamustine or CHOP-based therapy, largely first line for these early progressing follicular lymphoma patients, randomizing them to the 3 regimens you can see below. Next slide. And I think one of the things we've learned from this study is that while the patients are out there, they sometimes take some work to identify. So again, not to belabor previous points, but we've tried to make the study as lenient as possible in terms of enrolling patients. So patients can have received radiation, maintenance therapy. They have to have suspected or identified progression within 2 years. They do not have to be registered or on treatment within 2 years, meaning that they can be monitored for a period. Next slide. And part of this study that's very important is we want to better understand at a molecular level who these early relapsing patients are. So we are asking for slides from the diagnostic specimen, as well as at least a slide from the specimen where relapse was confirmed. However, there is flexibility. If there is if there are fewer than 10 slides available from diagnosis, patients can still be enrolled. If it's not safe to do a relapse biopsy, patients can still be enrolled. So again, lots of flexibility to enroll these patients. Next slide. So a bit of an update here. We've talked many times over, it's a challenging population to sometimes find and accrue. But again, they are out there. And interestingly, over the last couple of months, we've seen higher accrual than prior. 3 patients in August and September. I think we're up to 71 now. And notable contributions from City of Hope. MD Anderson just enrolled an additional patient. And interestingly, we've seen a lot of patients, probably half of all the patients enrolled are from community sites. So a lot of the smaller sites are able to find these patients and enroll some of these older patients, despite notable changes to the field like CAR T-cell therapy. Next slide. So a couple quick comments here just to comment where we're headed going forward. And I didn't put any slides in on this because literally Mike Hongley and I were emailing about this the last couple of days. We've talked about decreasing the power in this study and are prepared to submit an amendment using 80% power instead of 85%, reducing the sample size from 150 to 100 patients. And given that we're 3+ years into this study, this seems very reasonable. Knowing that this wasn't a dedicated phase 3 anyway, it does seem very reasonable to while we can, get this study done and analyze the results. And with that in mind, we were always planning to do an interim analysis at about 50% accrual, which is where we're at. Obviously, if we reduce the sample size, we'll be looking at about 75% roughly of the patients enrolled, and we're prepared to go forward with that. So hopefully this, we are starting to see the light at the end of the tunnel. We've seen an uptick in accrual. Please do what you can to help find and enroll these patients. We also looked at the correlative specimens bank so far, and there are a very large number. So we will be able to look at some very interesting molecular questions at the end of this trial. So again, thank everyone for their support, and we can move on. I think you're muted, Jonathan.
Sorry, if we're able to keep the trial going at about 3 patients per month, we'll finish this in a year. But it's going to take all the work of everybody. And as Dr. Smith put in the chat, I think this is one of the only studies in the world really dedicated to this population that was designated as the highest priority at the clinical trials planning meeting a few years ago. Any other questions for Dr. Barr?
Thank you.
All right, next, Dr. Stiff is going to discuss the Alliance study.
Good afternoon, everybody. Hope you can hear me. This is kind of like the anti-ALEX study because we're not doing well, and we're going to talk about some statistical changes that are in the works as well. So this is the Alliance 051301, which is co-chaired with the BMT-CTN. Babis Andreas is the PI from the Alliance at UCSF. So it's a randomized phase 3 trial of ibrutinib or norabrutinib during and following autotransplant for the ABC subtype diffuse large B-cell lymphoma. Next slide. So as everybody is well aware, patients with relapsed refractory DLBCL who've had salvage chemotherapy and have had a PR. So this is, again, as people start talking about Zuma-7, et cetera, CAR Ts, et cetera. This is the group of patients that would have gotten randomized to the standard chemotherapy arm and responded to therapy and then go on to transplant. So as we see those data come up in ASH, we'll have to look and see how did those patients do versus the CAR T cells. So it's an unanswered question, and hopefully people will still be encouraged to put patients on this trial. So again, the schema is as shown. Ibrutinib is given during the PrEP as well as for 12 cycles of monthly therapy daily, and then followed up. Next slide. Eligibility is, we've talked about this before, they can be progressed or refractory to first-line therapy, but no more than 3 prior therapies, no CNS lymphoma, and again, chemo-sensitive by local criteria. Next slide. The Amendment 6 now, it seems like it's only been a short period of time, but I went back and looked, and I think it was the summer of 2018. We changed from a nanostring requirement to the Hans algorithm. You send 6 unstained or stained slides to Cleveland Clinic, and within 5 days, and I'll validate that for you, you'll get your results back. So even as you're getting ready to start the PrEP regimen, you can get the results back. And again, does not have to be the relapse biopsy, can be the initial biopsy. Next slide. Again, BEAM or the SWOG CBV conditioning. Again, ibrutinib or placebo is given during the PrEP regimen and then after patients engraft. Next slide. Here's the actual maintenance, ibrutinib or placebo, 560 daily, continuously. start between day 30 and 60. You decide when your patient is ready to start the therapy. You can do 2 7-day drug holidays for reasons other than toxicity. The patient's just kind of had it and wants to take a little bit of a break. You can do that. Next slide. There's also a crossover. So if a patient progresses sometimes post-transplant and you think the patient might have gotten placebo, you can unblind them. And again, the trial will provide drug for up to 12 cycles. We are going to measure secondary progression-free survival as well. Next slide. So this was activated 2016, and a safety run-in was completed June of 17. As of June 2020, sorry, I don't know how that slipped in. Total registered and treated 84. Next slide. Now the initial N was 296. And the goal was to determine if we can see an improvement in outcome, 2-year progression-free survival of 17%, going from 50 to 67%. Because this trial has languished, and again, there's not been a formal amendment put out yet, but there's been discussions by the primary team and the NCI of alterations in statistical endpoints, as Paul Barr just talked about. This was originally to be a registrational trial, so it was very tight statistical analysis. And what's been agreed to with the NCI, I am told, again, not formalized, was that we could drop the number down to approximately 160. So essentially half of what we wanted. So given that we have about 85 patients, we're now 50% completed. So we need only 80 patients more to complete the study. And if we could get 4 per month, we'd be done in a year and a half. And again, the question is, will we, as of December, change how we manage these patients? I'm not so sure we will. Again, these are chemosensitive patients who are good transplant candidates. They're not going to get ICANS. They're not going to get a CRS. And many of these patients can actually be transplanted in an outpatient setting. Next slide. So that's it. I really hope that we can rise to the occasion and get those last 80 patients. I'm particularly looking at the top accruing sites. Actually, it's Duke and UCSF. In the top 10, there's only one SWOG center, and that's Rochester, of course. So keep going, guys. But again, we'd be really pleased. You know, I looked at the number of people that were participating in this conference call, and if everyone put one patient on, we'd be done with the study. So I'm fearful, however, that if this really doesn't move forward, that it's going to die, and we'll never have enough patients to really answer the question. So please consider opening the trial if you haven't, or activating the trial if it's possible, or of looking for patients to enroll on this trial. Thank you.
Thank you, Dr. Stiff. I think we'll probably move on just for time, but if there are questions, you can address them to Dr. Stiff. Let's move on to Dr. Till, who's going to talk about the ECOG mantle cell study.
Great, thank you. That's my disclosures. Next slide, please. So this is a frontline, well, not really frontline consolidation study of testing transplant versus no transplant in younger mantle cell lymphoma patients. So to try to mimic real world applicability, we're allowing any induction regimen essentially. And then so there's 2 steps to the process. The first step, we register patients and then send their tissue. The first step is really just to send their tissue to Adaptive to try to identify the clonal sequence in their tumor biopsy that we then look for later in the MRD test using the clonoSEQ assay. And so that can be done even before they even start induction therapy. It can happen during induction, ideally, or it could be after induction if needed. And so patients who then have a clonal marker then continue on the study. They get restaged with a bone marrow biopsy and PET scan after they're finished with induction. Those who get at least a partial response get the MRD peripheral blood test that's sent to adaptive. The people who get a complete response by PET and bone marrow and are MRD negative on the peripheral blood sample, that's the randomized population. So that MRD negative CRs, which is about 3/4 of all the patients. So this is what it's turning out to be so far. So those are randomized. They're stratified by MIPI-C and intensive versus non-intensive induction. And then they get randomized to an auto transplant or no transplant. And then both arms get maintenance therapy for 3 years. Everybody else, so the partial, if you get a partial response or are MRD positive or are MRD indeterminate, those patients all still stay on study. They just all automatically get this sort of standard of care treatment with auto transplant and rituximab maintenance. Next slide, please. So we're doing pretty well for accrual. We're targeting 689 total patients in order to try to get 434 randomized patients. And we've gotten 428 out of the 689 so far. A little over 3/4 of the patients are MRD negative. And SWOG is sort of bringing up the rear among the 3 main cooperative groups there, although I'm happy to say that over the last 6 months, I think we enrolled the same number as ECOG. So it seems like we're doing pretty well. And there's a few sites, Kansas, City of Hope, Seattle, and Utah that are on the leaderboard there doing well. So I appreciate everybody's help on that. There's been a little bit of appreciation that there's more patients than we had hoped are refusing their assigned treatment. And this is primarily affecting arm A, which is the transplant arm. So it's about 15% of the patients on that arm. So there's been some discussions about whether we might be able to, whether we need to increase the total number of patients on the study to try to not have the endpoints be compromised at all. So that's still under discussion. And I think we're going to ask for that, but it's, so whether or not we'll get an approval from CTEP, we'll find out. But as of now, these are the official numbers. Next slide, please. So we talked about this last time, and I'll just reiterate that we try to build in a little bit of flexibility in the COVID-19 era, where you can, if you want, delay some of or skip some of the doses of rituximab maintenance up to 4 missed doses. They still have to be made up at the end, but you can kind of delay if you need to for disease or vaccination. I will say that in light of the data that have come out in the last 6 months or so, showing that patients who received their vaccinations within 12 months of any anti-CD20 antibody therapy had essentially no antibody responses with their vaccines. I'm not sure skipping 4 doses is going to get the job done there, but if you want to do it, you can. I would also remind people to try, after that last cycle of induction therapy, please wait till their white count is recovered all the way to try to get as many genomes as we can in the denominator, because there's a minimum cutoff of 100,000 genomes that we need to be able to count the patient as truly MRD negative as opposed to indeterminate. If you're in a kind of a time crunch where a patient doesn't show up until after they've finished induction therapy already, and you want to kind of get things moving to either get them to transplant or maintenance therapy, you can send the diagnostic tumor tissue and the peripheral blood sample at the same time to try to kind of streamline the process, and Adaptive will sort of just hold on to the MRD test until the ID test has been officially run and shows a marker. And then, as I mentioned before, there are some patients who are refusing their assigned treatment. Those patients still, first of all, try to avoid that if you can by, I guess, kind of drilling it into patients that they really need to be willing to get either treatment. And if they're not, they just shouldn't go on the study if they feel so strongly about which treatment they're going to get. But I know it's challenging in a transplant versus no transplant study. There's a big difference there between the arms. But if they do refuse, they should stay on the study. So they don't come off study, they're still on study. We still continue to follow them, and we just will track them, their survival. And then eventually there'll be a, probably two separate analyses of, an intent to treat as well as an as treated analysis. So I think that's my last slide.
Excellent. Any questions for Dr. Till? Hildy is asking how many have refused their assigned treatment. I think we heard about 15% in that arm, right?
Yeah, it's about 15% in that arm. Yeah. So I think at the time it was like 150 patients. So whatever, 15% of 100, it's like 20 patients or something like that.
So again, this is another potentially practice-changing study that only the cooperative groups could do. And despite the challenge of having these discussions, it seems that many sites have figured out how you can talk to a patient and say you're either going to get a transplant or not. And probably, I think what a lot of people are saying is sort of transplant now versus transplant later. Is that probably the way that people are doing it?
Certainly, I think Tim Fensky, the PI of the study, that's kind of his approach. I haven't really used that in my spiel so much, and but it is certainly, I think, a reasonable thing. I think one key is to, if you can, I think it's easier if you catch them early in their disease course and talk to them about this, as opposed to if they've seen their local oncologist and it's been drilled into them that they're going to get a transplant, and then they come to the academic center and get a transplant consult, and they say, well, actually, we can do this study where we might not give you a transplant. It can be a little bit harder to deal with that.
Agreed. But this is clearly accruing well. It has good momentum. It'll be open another year or two at least. So still worth trying to get on board. Right. Yeah. Okay. Nicely done. Let's move on to Dr. Menguel's back yet again to discuss a Hodgkin's trial, which no, did I skip one? I'm sorry. It looks like Dr. Spurgeon's slides are next. So let's go with those. Steve, I think you're muted.
Okay, thank you.
Yep.
So the same principles apply here that Dr. Till mentioned with the MRD testing, but this is really the study you should think about when you see a newly diagnosed mantle cell lymphoma patient under the age of 70. As you all know, we're really trying to find a gold standard approach to get these patients into remission. And then to feed, this trial will feed into the 4151. So as you're talking to them about this, keep in mind the 4151 study. But essentially, this is a 3-arm study that builds on the experience using bendamustine Rituxan as well as Ara-C combinations, and then adding in a BTK inhibitor acalabrutinib to see if we can improve upon response. So here you can see a randomization. There are these 3 arms, and cycles 1, 2, 3 is BR, and cycles 4 through 6 is cytarabine Rituxan. So that's 1 arm, and that is really the control arm in this study, and the other 2 arms are compared to that. And we have arm B, which incorporates the Ara-C BR approach, but also adds in acalabrutinib. And importantly, first 3 cycles, acalabrutinib is continuous on days 1 through 28. And then with cycles 4 through 6, of course, given the thrombocytopenia, it is interrupted between days 8 and 21 and resumed on day 22 through 28. And then arm C is trying to understand, can we, and we had to work pretty hard to get this into the protocol. So I think we're proud of this as SWOG, because I think we really drove this home, is how does an outpatient regimen just with the BTK inhibitor perform? I think, again, the primary endpoint is MRD negative CR, and we're trying to see which arm leads to the highest rate of MRD negative CR. The same principles apply with the tissue. I think importantly that patients that have their tissue or slides submitted here for clonoSEQ testing can use that same tissue for the 4151, that same result. And we really want to make it as easy as possible to enroll these patients. So patients with non-measurable disease can be enrolled as well. There's a lot of flexibility as far as, inpatient cytarabine versus outpatient cytarabine, using biosimilars. That's been a question as well. And I think the key thing here is that the study got off actually to a pretty slow start, but here you can see it's picked up. So we're about halfway home here, and I think really things have picked up. We are starting to make a little bit more of a run. ECOG is enrolling a lot of patients, but we're next in line. And here you can see the usual cast of characters here, but University of Chicago has jumped into the mix as well with the leaderboard, so to speak. So again, I think this is a really important study for this population, especially adding in the non-intensive arm to see if that is an adequate approach for these patients. So that's, I'll take questions.
Any questions or comments? This is a very straightforward, study, I think, and also, as Dr. Spurgeon said, very nicely flows into the other study. So it's a way that people can, kind of get a whole treatment plan dictated by 2 clinical trials. Maybe I'll just ask Dr. Smith, since a lot of patients have enrolled from Chicago, any experience you want to just say about on this study?
Yeah, I would just say that I find this to be one of the easiest trials to accrue to. Patients don't know exactly what they want to go on. All of the arms make sense. We talk about cytarabine, about BTK inhibitors, and it's been really straightforward.
All right. Thank you. And then now I think we move to the Hodgkin trial. Oh, not quite. Okay. So this one, Dr. Villarupalli, I think is on hold, right? But I'll let you kind of summarize where we are with this one.
Yep, this is, as you said, Jonathan, currently closed for enrollment, but it's, as everyone knows, a randomized study comparing chemoimmunotherapy plus or minus venetoclax with double hit and double expressor lymphomas. Next slide, please. So this is the scheme of the study. Untreated double expressors get R-CHOP versus R-CHOP plus venetoclax. Untreated double hits, the initial study was DARE-POC plus or minus venetoclax. Next slide, please. So the current updates, the double hit arm is now closed for enrollment. The plan is to proceed for phase 3. So look for updates in the next few months about phase 3 reopening. Where this is pending is event-based data analysis and redoing the initial null hypothesis. So when the study was designed for the double expressor arm, the null hypothesis was 24-month PFS of 45% versus 65%. But some of the recent studies had showed some updated PFS rates. For example, the GOA study, PFS was 62% at 5 years. So the independent statistician is reworking. So hopefully we'll get the statistics plan updated and get this reopened for enrollment, hopefully by the end of this year.
And I don't mean to put you on the spot. So I'll preface this by saying that, are you familiar with the ASH abstract on the double hit component?
That's my next slide.
Oh, didn't mean to interrupt. Perfect. See, I was setting you up.
No worries. Next slide, please. So the double hit arm, as many knows, has been on hold for enrollment almost all this year. because of the safety signal, we had 5 deaths from septic shock secondary to neutropenia in the dose-adjusted REPOC plus venetoclax arm. There was an abstract submitted for ASH with the toxicity data, so look out for that. But it's mainly prolonged neutropenia in most patients, although a few patients it was early on neutropenia and septic shock. So this arm, as it has been written in the initial study, is not going to go forward. There have been some ongoing discussions with the NCI CTAP, and the current plan for this double hit arm is redesigning of this arm with statistical analysis is pending. And the way it's going to move forward is DA-REPOC versus R-CHOP plus venetoclax. So this is pending statistical analysis. The arm will reopen, but that will take a little while longer. And then the toxicity data will be presented at ASH for the neutropenia and the septic shock, and predominantly in the venetoclax combination arm.
So again, I think this highlights the importance of these clinical trials. There is a manuscript that just came out on the phase 1 of the EPOC and venetoclax, not really showing those safety signals. So it again emphasizes the importance of then broadening and doing this in the group. And I think when you see the data that's being presented at ASH, you'll realize why that double hit harm had to close. So I imagine we'll probably have to get an update at the next meeting. But if we'll certainly send out a notification if there's going to be one of these arms reopening. But my guess is it's probably still a few months away. Is that your sense?
So when I spoke to Jeremy last week, it's still pretty early on. CTAP just approved this last month. The independent statistician hasn't had a chance to work on the double hit stats yet. It's still pending with the double expressor stats. So I think my guess is probably at least January is where we're looking for the double hit arm. I think the double expressor arm probably sooner than that.
I think one of the positive things that has come of this is the demonstration of the ability to accrue to these studies in both arms that has been quite robust, particularly double hit, which I think we recognize as an uncommon but generally poor outcome lymphoma. And there was some concern when the study was designed that people may not recognize this right away in time to put patients on study, but clearly that was not a barrier. And in fact, the accrual there was so robust that there was talk about potentially expanding the study before the toxicity signal came out.
Mm-hmm. And this is still a pretty needed space. There's not much data and no other competing studies at this point. So yes, I think enrollment won't be an issue once it reopens. And the fact that it's R-CHOP plus venetoclax, which worked out well for the double expressor arm so far with not a toxicity signal, is a pretty good comparator.
All right. Very nicely done. Did you want to say something, Soni?
No, I agree. I think, when we try to have these targeted agents, this was sort of unexpected to have this degree of toxicity, but it's really, great to be able to reopen it with a sort of, maybe it is the chemo that can be de-escalated. Mm-hmm. That's good.
Okay, I think Dr. Stevens is coming back for an encore now.
Thanks.
And you can do both of yours, I think.
Yep. Thanks for letting me present some studies that are based out of the leukemia group here, as I suspect a lot of the people who are accruing patients to these studies are actually in our lymphoma group here. I'm going to be presenting S1925, which is a randomized phase 3 study of early intervention with venetoclax and obinutuzumab versus delayed therapy with venetoclax and obinutuzumab in newly diagnosed asymptomatic high-risk patients with CLL, the EVOLVE CLL study. And next slide. The study schema is depicted here, and as I mentioned, the people who are eligible or newly diagnosed, asymptomatic, a CLL IPI score of 4 or higher, and or complex cytogenetics. These people are being stratified between high risk and very high risk on the CLL IPI score and randomized 2-to-1 to early versus delayed venetoclax and obinutuzumab therapy. The primary endpoint of the study is a lofty overall survival with a planned accrual of 247 patients. The secondary endpoints and translational endpoints are listed here. You want to go to the next slide. The status of this study currently is we were officially open to accrual in December of 2020. Right now, our current accrual is 11 of the planned 247. We had some, a little bit of uptick right when the study opened, and it slowed down a little bit, I think, given the recent COVID spike. But there are a lot of stipulations on the study that allow for some delays due to COVID-related issues. And so I would ask that you all please still consider enrolling your patients in this study. Go on to the next slide. We just passed through amendment number 1. This does allow for 3 days of prednisone at 50 milligrams prior to the first obinutuzumab infusion in order to prevent infusion reactions. It clarifies some discrepancies in obinutuzumab infusion rates. It provides guidelines for patients who develop COVID-19 infection while on study, and it clarifies some timing between registration, screening, and treatment. And so, like I said, this study is open. It's a big benefit to patients because it does provide the drug to patients, whether they're on the early versus delayed arms. You can go on to the next slide. I'm also going to be providing an update on behalf of Dr. Advani at Cleveland Clinic about the S1905 study, which is a phase 1 study of OBI 3424 in patients with relapsed or refractory T-cell ALL or lymphoblastic lymphoma. Next slide. OBI 3424 is a nitrobenzene prodrug of a nitrogen mustard that can be selectively cleaved in the presence of AKR1C3 enzyme, which is an enzyme that is expressed at higher levels in T-cell lymphoblasts compared to other cell types. You can see the eligibility for the study listed here on the right, relapsed or refractory T-cell ALL or LBL. If they have lymphoblastic lymphoma, they must have greater than 5% lymphoblasts in the bone marrow or peripheral blood. Prior nelarabine is not mandated. The phase 1 portion of the study is a dose finding. It's a 3+3 design, and you can see the dose levels here. The primary objective of this part is, of course, to find the MTD, and then the phase 2 portion will be an expansion cohort with a primary objective of complete response rates. This is an intravenous therapy. It's given days 1 and 8 of a 21-day cycle. And responding patients may receive up to 17 cycles, and partial remission by cycle 4 previously was required, but in a new amendment, you're allowed to continue the patients if you feel that they are deriving clinical benefit. And patients may proceed to allogeneic bone marrow transplantation if a CR is obtained. You go to the next slide. And this is a fairly limited population. I think they have had 7 accruals so far, and they are on the second dose level. They did not reach a DLT or MTD in the first dose level. And the next slide. And as I mentioned before, patients can continue on therapy even if PR is not reached by cycle 4. And then a new amendment is being planned for submission addressing blood count threshold requirements for ongoing cycle administrations and guidance for dose toxicity, dose adjustments for toxicity and prohibited concomitant meds. And again, there's no clear toxicity signal so far and no DLT event recorded. And that's my last slide.
Any questions for Dr. Stevens on either of those studies? I think the CLL study is an important study. Our approach of deferring treatment is based on studies from the 1980s, both in follicular lymphoma and in CLL. And in this new era, now that we can risk stratify patients, particularly in CLL so well, it's really an appealing question to ask. So encourage everybody. And it also, I think, helps formalize an approach on risk assessment when you meet patients, which is the current standard of care. And, that way you can communicate with the patient that because of a certain level of risk, you're eligible for a trial.
Yep, and just don't forget that you have to check beta-2 microglobulin and TP53 mutation FISH in order to stratify these patients.
Debbie?
Can you go ahead, Dr. Barr?
Quick question. If we know there's many different ways to measure risk, and as we screen every new CLL patient, I sometimes have a hard time finding a high enough risk CLL IPI, a CLIPI score, but find tons of mutations. If there are difficulties enrolling, have there been any discussions about expanding the definition of higher risk?
We have not yet, and I think we're being given some leeway, just given this population is a little bit difficult to enroll during the COVID pandemic. But I think that's a great point, looking at other mutation as definition for high risk. The other thing, the patients do have to be within 1 year of diagnosis, and I think that that's another area that if we could get agreement to prolong that period of time, that would help with enrollment as well.
That's a little hard though, because then you have people who have been watched for a really long time and they may have already declared themselves to kind of be in one direction or another, I would think. So
maybe just a COVID 2-year period.
Yeah, I'm just, I think that it's just, you're trying to intervene biologically at some point. So some of this is going to take a little bit of time, I expect, because again, not every site has been running all of these tests on every new patient who otherwise wouldn't require treatment. And now there's a reason to do it. And I think anytime we define a new group for study, it takes a little bit of time to ramp up. So it's good to keep presenting this.
Yeah, and I think even when people do need treatment, it's not yet routine for all of those parameters to be tested. I mean, there is data that despite support for mutation status, et cetera, that it's not routinely done. So
right,
we have to keep at it.
All right. We made it through with 3 or 4 minutes to spare. So I don't think we have any more slides. We can probably take that down. And I'll just thank everyone for their attention. We had very good retention of people staying online during the whole meeting. I think it gives you a sense that we've really evolved from having only a couple of studies to really having a broad profile. And the good news is I think we're going to have some new studies to talk about again in the spring. And I think we're all hoping that will be in San Francisco. Dr. Smith, any parting words?
Just that I'm really proud of, how far this group has come together. There's just been, a tremendous amount of activity, a lot of hard work, a lot of careers growing and blossoming through this mechanism, and all to the benefit of our patients as we get these trials done. And as Jonathan just said, we've got even more, on the horizon. So thank you to all our speakers and our committee, and thanks to the to the group that's on this call for putting your patients on these trials and really being part of the process as we move forward.
Last person I'll just call on. Hildy, do you have any announcements from the patient advocate perspective?
Well, I would just like to tell the people that are participating today that are at our study sites to thank them. This lymphoma working group really enables my engagement as an advocate in all of these studies as they're being developed and then once we're accruing. I'm very pleased, from a patient advocate perspective and excited about all of these opportunities for patients and really excited about the activity of our committee in particular and the studies that we've been opening and will be opening soon. So I encourage you all to participate. We have added we have 7 out of 10 community advocates that are also on our patient advocate committee now that represent diverse groups and also can contribute to development of these studies. So that's all I have for now. And again, thank you.
Thank you. You corrected me. You said spring meeting will be in Seattle. I think wherever it is, if we're allowed, we'll be there.
Yeah.
Even if it's in Rochester, New York, people will come this time.
There's a limit. There's a limit.
Dr. Brem would come. I know she would be there. All right. Thank you all. And
Thank you.
talk again soon.